Good afternoon, everyone. Welcome to the day 4 of our interdisciplinary student webinar series. We arrive at today's session following 3 highly successful webinar days. We began our journey by exploring the molecular fundamentals of cancer biology and neuroscience, and most recently, we have even examined the shared mechanisms across human diseases and the core foundations of drug discovery. Today, we take a significant step forward by focusing on the bench to bedside transition and the digital revolution in science. Our fourth session is structured around two interconnected themes designed to bridge the gap between theory and application. Theme 7 constitutes the translational drug discovery. Here, we will explore the critical journey of bridging the laboratory research with clinical innovation to ensure that the scientific breakthroughs reach the patients who need them the most. Again, the theme 8 responds to bioinformatics and computational biology. We will delve into how computational tools are transforming modern biomedical research, allowing us to accelerate discoveries through the data-driven insights. As we broadcast live on Google Meet as well as YouTube Live, we are excited to showcase lectures from our dedicated student presenters alongside expert perspectives from our international professors as well. We encourage all participants to engage actively in the discussions as we navigate these complex but vital topics. To officially open today's proceedings and set the stage for our first theme, it is my distinct honor to welcome Dr. Sukant for the opening remarks.
Teaching is an important part of growing up as a scientist, to be an impactful researcher, impactful communicator. I wish the youngsters presenting today to a wider audience than just their research colleagues all the best. Go ahead and have a great day. Thank you.
Thank you, Dr. Sukant, for your wonderful words. And now I would like to begin the first session of the theme 7 with Jasmine. Jasmine Steffi is a biotechnology researcher with interests in translational medicine, immunotherapy, computational biology, and biomaterials. Her work focuses on bridging fundamental biological research with innovative therapeutic applications to advance modern healthcare. Welcome, Jasmine.
Greetings everyone, it's a pleasure to be here today. I am Jasmine Steffi and I'll be presenting on the title Translational Immunotherapy from Bench to Bedside. This title exactly captures what I will be talking for the next few minutes. It's not just the science but the process of turning science into therapies that saves life. This is a field that has genuinely changed what oncology can offer patients. And I think by the end of the session, you'll understand both the extraordinary process that we have made and the significant amount of work that remains ahead of us. Let's get started. First, let me establish what we actually mean by translational medicine. The phrase bench to bedside has become something of a cliche, but the underlying concept is profound. The traditional medicine is a systematic effort to close the gap between the fundamental scientific discovery and the therapy that a patient can actually receive. For a too long time, basic science and clinical practices operated in two different parallel universes. A researcher would publish a compelling finding about cellular mechanism, and it would definitely sit on this papers alone. And translational medicine says those two worlds must actively talk to each other. You can see on the slide like the four pillars that defines the integration, that is basic science and followed by biotechnology, clinical medicine and pharmacology. None of these work in isolation. to be honest. And crucially, the arrow doesn't go only one way. The reverse translational, where clinical observations feed back into the lab, is also very important. A patient who responds unexpectedly well to a drug teaches us something fundamental about the biology, and the practical stakes are real. Bridging translational gaps can cut drug development timelines by up to 30%, while improving trial success rate. In oncology, where time costs lives, that truly matters. This slide speaks about immunotherapy. Let me begin with what is immunotherapy and why has it captured so much of attention. At its core, immunotherapy is the idea that we should harness that the body's own immune system rather than bypassing it to fight a disease. This stands in contrast to conventional approaches like chemotherapy, where the treatment attacks cancerous cells directly, but as we all see shortly, at significant cost to the healthy tissues. Immunotherapy is a single thing. It is a broad family of strategy. Three major disease areas are covered here, like cancer, autoimmunity, and infectious biology. But today we'll focus primarily on oncology because the progress has been very dramatic in this side. The five modalities that I would touch today are monoclonal antibodies, checkpoint inhibitors, CAR T cell therapy, cytokine therapy, and cancer vaccines. Each works differently, targets different parts of the immune circuit, and has its own set of clinical applications. What they share is a fundamental philosophy that the immune system do what it's supposed to do and get out of its way, or give it a nudge in the right direction so it can perform better. How this immunotherapy, or why this immunotherapy has revolutionized medicine. To understand why immunotherapy is such a big deal, it also helps us to understand what came before it. For decades, the oncology toolkit consisted of surgery, radiation, followed with some chemotherapy. Each of these attacks the tumor directly, but the problem still exists. They are blunt instruments. Chemotherapy doesn't distinguish between rapidly dividing cancerous cells and rapidly dividing healthy cells, which is why patients lose their hair, experience bone marrow suppressions, and various and innumerable number of side effects. The table on this slide, summarizes the contrast. Look at the two characteristics of conventional therapy and immunotherapy. Resistance and durability. Conventional chemotherapy frequently encounters drug resistance through mutation and often doesn't last, but immunotherapy can generate durable responses, sometimes years long remission. Because of immune memory, the immune system remembers it fought this tumor and stands ready to respond again. This is what makes immunotherapy qualitatively different. We have seen patients with metastatic melanoma, a disease that was essentially a death sentence a decade ago, achieve what appeared to be a functional cure. That's what's supposed to be possible. Before we go further into the therapies themselves, I want to spend a moment on the immune architecture that underpins all of this. Because you cannot rationally design a drug without understanding the system that you're targeting. The immune system has two arms: the innate immune system, that is the first line defense, it sees danger, sounds the alarm. The adaptive immune system is lower, but it learns and remembers. The T and B lymphocytes undergo clonal selections, meaning that when they encounter a specific antigen, they expand into an army of cells targeting exactly that threat. For drug discovery, three concepts are critical. First is antigen presentation, and followed with T cell activation and then cytokine signaling. In antigen presentation, the tumor cells display fragments of their own proteins like MHC molecules on their surface, like flags identifying themselves on the immune system. One of the first things tumors do to evade immunity is hide those flags, and that tells us a lot about where to intervene. Second, the T cell activation requires two signals. Signal 1 is the T cell recognizing the antigen, and the signal 2 is a co stimulatory signal confirming this is a genuine threat. If signal 2 is missing, the T cell doesn't just fail to activate, it becomes energetic. Checkpoint inhibitors specifically target this axis. Third is the cytokines. These are the soluble messengers that coordinates the immune response, amplifying, suppressing, and recruiting the immune system. They are among the most powerful levers we can pull in immunotherapy. Now, here are the adversarial realities that makes the immunotherapy necessary and challenging in equal measures. Tumors are not passive targets, they actually evolve with time. There are four major evasion strategies that I want to highlight here. First, tumor can downregulate antigen presentation. They essentially stop displaying those identifying red flags, rendering themselves invisible to cytotoxic T cells. Second is tumors exploit immune checkpoints like PD-1, PD-L1 axis. In a canonical example, tumors upregulate PD-L1, which engages PD-1 on T cells and delivers a stand-down signal. The T cell doesn't kill, it exhausts. The third signal is tumors secrete immunosuppressive cytokines like TGF-beta and IL-10, creating what we call as an immunologically cold environment, a tumor that excludes effective immune response. Fourth is tumor microenvironment remodeling. Perhaps it is the tumor remodeling the stromal structures by recruiting polarized macrophages that actively suppresses effector immunity. Understanding these mechanisms isn't just academically interesting, each one represents a potential drug target. Each one of these field has essentially been cataloging in the enemy's playbook and then designing interventions to counter each move. Immune checkpoint inhibitors. This brings us close to checkpoint inhibitors, arguably the most transformative advances in oncology in the past 2 decades. The concept is elegant. If tumors are exploiting braking signals to shut down immune responses, block those brakes. Checkpoint inhibitors are monoclonal antibodies that bind to inhibitory receptors like PD-1, PD-L1, or CTLA4 and prevent them from delivering suppressive signals to T cells. The result: exhausted, energetic T cells are rejuvenated and return to killing tumor cells. The tumor major targets are shown here, that is PD-1, PD-L1, and CTLA4. Operate primarily, this first PD-1 and PD-L1 operate primarily within the tumor itself. The next one acts at the level of T cell priming in the lymph nodes, which is why combining anti-PD-1 and anti-CTLA4 has synergistic effect. Let me highlight the three landmark approvals. Ipilimumab, the first checkpoint inhibitor approved in 2011 for metastatic melanoma, proving for the first time that the immunotherapy could extend overall survival in diseases. And pembrolizumab, approved across 15 or more indications, the first ever drug approved based on a molecular feature of a tumor rather than where the tumor is located. And nivolumab, which is a combination of the previous both, has produced survival outcomes in renal cell carcinoma that shape the entire first line treatment paradigm. Monoclonal antibodies deserves their own section because they underpin so many different immunotherapy strategies, not just checkpoint inhibitions. And monoclonal antibody is an engineered immunoglobulin that is designed to bind to a single epitope with very high specificity. There are now around hundreds of FDA-approved monoclonal antibodies across oncology, autoimmunity, and infectious diseases. They work through several mechanisms from direct neutralization of the target (ADCC), where the antibody recruits the NK cells to kill the antibody-coated tumor cells, and complement activations. Two milestones that stands out are rastuzumab and Herceptin that transformed HER-positive breast cancer from the worst subtype into manageable disease. It targets HER2, a growth factor receptor overexpressed in about 20% of the breast cancer, and blocks the downstream signaling that drives tumor proliferation while also triggering ADCC. Rituximab, did the similar thing for B-cell lymphomas, targeting CD20 on B cells and producing clear overall survival benefits over chemotherapy alone. And the field has continued to evolve from monospecific to bispecific antibodies, antibody-drug conjugates, and Fc-engineered formats are expanding every day. CAR therapies. If monoclonal antibodies represent precision targeting, CAR-T therapy represents something altogether more remarkable: a living drug. This concept is, we take a patient's own T cell, genetically reprogram them to express a chimeric antigen receptor that targets a tumor-specific antigen, expand them in labs, and reinfuse them. These cells can proliferate inside the patient and traffic to tumor sites and persist as a memory cells, providing long-term surveillance. The CAR construct has four functional components: an extracellular antigen binding domain, essentially a targeting missile, a hinge region providing flexibility, and a transmembrane anchor, and a crucial intracellular signaling domains that provide both signal 1 and signal 2 for T cell activation. Getting that co-stimulatory component right, that is CD28 versus 41BB, has major implications for T cell persistence and exhaustion. The pivotal moment was 2017, a first ever FDA approved CAR T product for pediatric B cell was in the market. The Eliana trial achieved complete remission rates of approximately 81%. For children who had relapsed or were refractory to conventional therapy, this was transformative. Since then, approvals have expanded to diffuse large B cell lymphoma, multiple melanoma, and follicular lymphoma. Cytokine-based immunotherapy. Cytokine therapy is in some way the oldest immunotherapy approach, and it illustrates both the power and peril of directly manipulating the immune signals. IL-2 was among the first FDA approved immunotherapies administered at high doses. It can drive robust T cell and NK cell expansions and produce durable, complete response in melanoma and renal cell carcinoma. The problem is that those responses come in only 5 to 9% of the patients, and the toxicity profile is higher, causing hypertension, multi-organ stress, limits its use to highly specialized centers with extensive monitoring capacity. Interferon alpha has a different profile. It enhances tumor visibility by upregulating MHC class 1, activates innate cytotoxicity cells, and has direct anti-proliferative effects. It was historically used as a hairy cell leukemia (CML) and melanoma. While it's been largely seen, it remains relevant in combination regimens. Cancer vaccines. Cancer vaccines occupy a fascinating space in this landscape. They are the most conceptually intuitive approach, but also the most challenging to execute. There are two broad categories: preventive vaccines target oncogenic pathogens before malignant transformation occurs. Example, HPV vaccine is an outstanding success story, here targeting the strains responsible for approximately 70% of the cervical cancer, with efficacy exceeding 90% in vaccine-naive individuals. This cancer prevention is done at a large-scale population. Therapeutic vaccines are administered after diagnosis and aim to amplify tumor-specific immunity. Spural T for prostate cancer was the first FDA-approved therapeutic cancer. The benefit is modest, a 4-month overall survival improvement, but it approved the principle. You can train the immune system to target a tumor antigen in an established cancer. The cutting edge is personalized neoantigen vaccine. The tumor-specific mutation generate novel peptide sequences, and using whole exome sequencing and computational prediction of MHC binding, we can design a vaccine uniquely targeting a patient specific tumor. Translational drug discovery pipeline. Now let's step back and look at the pipeline through which all of these agents passes, because understanding where things fail is important as understanding the science itself. Statistics says that approximately 95% of oncology drug candidates that succeeds in preclinical models fail in clinical trials. That's not a pipeline problem, that's a translational problem. Why does this happen? Most tumor models, however useful, do not replicate the immunological complexity of a human tumor microenvironment. In vitro assays cannot capture pharmacokinetics variability, and patient's heterogeneity means an effect seen in small, selected population may disappear in a broader trial. There are 4 critical inflection points where failure cluster, and these are like target to lead. If the target biology is underpowered, phase 2 will fail. Preclinical to phase 1, species specific toxicity, pharmacokinetics, pharmacodynamics mismatches. Phase 2 to phase 3, inadequate biomarker stratification dilutes efficacy, and at regulatory submission, the FDA increasingly expects companion diagnostics to be developed in parallel with precision agents. The solutions are patient-derived organoids, humanized mouse models, adaptive trial designs, and biomarker-derived stratification. The role of biomarkers in immunotherapy. Biomarkers are, I would argue, the single most important enabler of precision immunotherapy. They answer the questions that matters the most. Which patients will respond? How is the treatment working? And what toxicity risk can we anticipate? 3 functions. First, predict patient's response. Predicting response like PD-L1 expression guides anti-PD-1 eligibility in multiple tumor types. High tumor mutation burden, 10 mutations per megabases or more, predicts response across histologies and supports the first tumor-agnostic FDA approval. MSI-H status is a prototype predictive biomarker with response rate above 40% in certain colorectal cancer. Second is monitoring therapy. Circulating tumor DNA (cTNA) can detect molecular responses week before a CT scan shows any changes. Serial ctDNA measurement can also identify emerging resistance mutations in real time, enabling real interventions before clinical progression. Third, reducing adverse effects. Immune-related adverse events are a major challenge in checkpoint therapy. Baseline autoantibody panels, thyroid function tests, inflammatory cytokine signatures can flag patients at high risk of pneumonia and several other diseases. The direction of travel is composite biomarker signatures, combining genomic, proteomic, and immune data. AI and computational biology in immunotherapy. Artificial intelligence is accelerating every stage of this pipeline, and I want to be specific about where. Drug target predictions (ML) models trained on pan-cancer multiomics datasets can identify synthetic lethal interactions and context-dependent vulnerabilities that no hypothesis-driven approach could never efficiently do. Neoantigen identification. Deep learning can predict MHC peptide binding affinities with near experimental accuracy, enabling rapid nominations of high priority vaccine targets from sequencing data. AlphaFold 2 deserves special mention. The ability to predict protein 3D structure form sequence with atomic level accuracy has unlocked structure based drug design for previously undruggable targets, a genuine shift for drug discovery. Clinical response prediction, multimodal interactions like radiology, digital pathology, genomics, and clinical variables are increasingly reliable in predicting who will respond to immunotherapy and who is at risk for severe adverse event. Clinical trials in immunotherapy are where the science meets the patients actually, and the unique biology of immunotherapy has required fundamentally redesigning how we run them. The phase structure remains same almost. Phase 1 for safety and dose finding, phase 2 for preliminary efficacy and biomarker validation, phase 3 for the large randomized evidence that supports regulatory approval. But immunotherapy has revealed that limitations of design built for cytotoxicity chemotherapy. Three innovation that stands out: adaptive trial designs allow real-time modifications of dose, arm allocation, and patient selection based on data. Critical when you are dealing with a biomarker stratified population. Basket and umbrella trials enroll by molecular signatures rather than the tumor location, enabling efficient evaluation across histologies. And immune modified response criteria accounts for pseudoprogressions, where a tumor appears to enlarge briefly as immune cells infiltrate before the actual response occurs. The challenges are equally real: patients' heterogeneity, delayed and unusual toxicities, patients' tumor heterogeneity that makes a single biopsy an imperfect proxy for the entire tumor immune landscape. This is followed with the challenges. I want to be honest about the challenges because they are very significant and they should shape our research priorities tomorrow. Scientifically, tumor heterogeneity remains a fundamental obstacle. Tumor immunotherapy may eliminate sensitive subclones while enriching resistant populations, and tumor continue to evolve under selective pressures, upregulating alternative checkpoints like TIM3, LEG3 as blockade becomes a selection pressure. Economically, the numbers are sobering. The CAR-T therapies carries price list from millions of dollars sometimes. Perfusions, checkpoint inhibitors frequently cost a lakh of dollar per year, and these treatments are transformative but are not accessible to vast majority of global population. Advanced immunotherapy currently requires infrastructure, starting from genomic testing platforms to facilities and specialized oncology center. Ethically, CRISPR-based immune cell engineering raises questions about off-target genomic edits and long-term immunological consequences that require rigorous preclinical characterization before broad clinical adoption. Let me bring this to life with a case study that presents the most compelling proof of concept for cancer immunotherapy: metastatic melanoma. Before 2011, the picture was bleak. Our standard of care was just chemotherapy with objective responses rates from 10 to 15% and virtually no durable complete response. IL-2 therapy could occasionally produce durable remissions, but with life-threatening toxicity. The median overall survival was 6 to 9 months from diagnosis to stage 4 disease. 5 year survival was under 5%. The disease was considered essentially incurable. Then these checkpoint inhibitors arrived. Imluvab in 2011 was the first agent to demonstrate a survival benefit. The subsequent introduction of nivolumab and pembrolizumab alone and in combination transformed the survival curve in a way that's genuinely remarkable. The bar chart on the slide says the story. 5 year overall survival climbed around 5% overall to 52% with combination with nivolumab plus ipilimumab. Complete response rates of 10 to 15%, many of which appear functionally curative. 10-year follow-up data from the original trials shows plateau in the survival curve, a trail of long-term survivors that simply didn't exist before. Let me point the session into five different core takeaways. One, immunotherapy has fundamentally transformed oncology. Durable, complete response that were previously considered unattainable are now clinically achievable. Two, translational research is the bridge. Rigorous bench to bedside work with mechanistic validation, iterative feedback between lab and clinic is what produces the impact. 3. Biomarker and AI-driven precisions without validated predictive biomarkers and computational tools to analyze complex data, we cannot deliver the right therapy to the right patients. 4. Major challenge still remains resistance, toxicity, and cost. Progress requires both scientific innovation and structural healthcare reform. 5. The future is very personal. Every converging technology, CRISPR, neoantigen vaccines, organoids, and nanotechnology is steering us towards a paradigm where the treatment is tailored to each patient's unique tumor biology. I would close with a reflection rather than a summary. The trajectory of cancer immunotherapy from foundational checkpoint discoveries by Allison and Honjos, recognized with Nobel Prize in 2018, to durable remissions in disease that were incurable a decade ago is one of the most compelling stories in modern medicine. It is what rigorous, collaborative, patient-centered translational science can achieve when it operates at its best. The roads ahead are precarious. The next generations of researchers and clinical scientists to solve immunotherapy's hardest remaining problem, that is overcoming resistance, making these therapies globally accessible, and engineer safer and more potent cellular agents, and also design trials that generate knowledge at the speed of discovery. The tools are available and the patients are waiting and the science is ready to. Thank you for your time.
Thank you so much, Jasmine, for your insightful lecture on immunotherapy and the translational aspects. Now, I'm going to welcome our second presenter on the lineup, Poshali Bose. She has done her master's in um, molecular biology and biophysics and now currently working with us. Welcome, Poshali.
Hello. Hello.
Yes,
Am I
you're
audible?
audible. Yeah, go ahead.
Hello.
Hi,
Yes, you're
Poshali.
audible.
You're audible, Poshali.
Hello.
Hi, Poshali. You're audible. Go ahead. You're audible. We can hear you.
Okay. Hi, everybody. I hope I'm audible.
Yes, yes, you're audible. Go ahead. Kindly go ahead.
Sorry. I'm sorry for the delay. Please give me 1 minute. Hello, everybody. My name is Paushali Bose. Good afternoon, everybody. So it's my privilege to present today on translational drug discovery, bridging the research and the clinical innovation. I'm sorry. I'm sorry, my laptop is hanging as well. Okay, so modern biomedical science has generated extraordinary advances in molecular biology, genomics, and therapeutic technology. So, yet, one fundamental challenge remains: how do we efficiently convert the scientific discoveries into therapies that truly improve patient outcomes? Translational drug discovery addresses this challenge by creating a structured pathway from laboratory research to clinical application, integrating biology, technology, medicine, and patient centered innovation. So, today I'm going to talk about all these contents that have been mentioned here. So let's talk about the core concept and importance of translational drug discovery. Despite major scientific breakthroughs, a significant proportion of promising discoveries never reach patients. So the gap between the discovery science and clinical implementation is always often referred to as valley of death in drug development. Translational medicine aims to bridge this gap by connecting basic research, preclinical development, clinical investigation, regulatory science, and healthcare implementation into a proper continuous translational continuum. Importantly, translational success is not achieved by a single discipline. It requires coordinated collaboration between academia, industry, clinicians, regulators, and patients themselves as well. So here comes a lot of W here, which includes what, why, who, when, where, and how. So every successful translational program usually begins with a clinical meaningful question. What unmet patient need are we trying to address? From there, translational strategy becomes a highly structured process. Scientific objectives, target validation, dose rationale, biomarker plans, and clinical endpoints, they all must align with the intended therapeutic goal. So equally important that it is multidisciplinary ecosystem where integrating expertise all the fields, include especially emphasizing on the pharmacology, clinical science, biomarker development, and regulatory strategy. So, a robust translational roadmap ultimately serves one purpose: reducing the uncertainty while maximizing the probability of clinical success. As at this point, we all are aware of high attrition rates. So let's next talk about the translational pipeline. Though we have already talked about it in last couple of our talks, but we'll just go into it briefly. So once the translational framework is established, development progress through a series of these interconnected stages, which begins with the target identification and hit discovery, followed by lead optimization, clinical validation, and regulatory approval. However, translational drug discovery is not a simple linear pathway. It is highly iterative, and data generated during early clinical and development frequently reshape this biological hypothesis, biomarker strategies, and even the target assumptions established during the preclinical research. So this bidirectional learning process is central to the modern translational science. So let's see what is the success status of this translational research. So traditionally, we know that drug development success was measured primarily through regulatory approval, which usually takes 10 to 15 years. Today, translational success is evaluated much earlier and much more comprehensively. So key matrices include reduction of attrition rate, reproducibility of the findings, and biomarker validation, which we can see from this picture how the success rate has changed over the course of time. Progression into the first human studies and the ability to identify failure risks before expensive late stage trial. So, in translational medicine, recognizing failure early is often as valuable as achieving success late. So, let's talk about target to candidate, that is, the preclinical transitions. As we have already talked about these things, we'll not go much deeper into it. So, one of the first major translational challenges is transforming the biological insight into a valuable therapeutic candidate. So, advances in genomics, proteomics, system biology, and computational modeling now allowing us to identify highly diverse, disease relevant targets with increasing precision. Following target validation, modern discovery platforms, including high throughput screening, structure guided design, and fragment based discovery, for example, trisozumab, and AI based assisted virtual screening, accelerate this hit identification and optimization. So, candidate molecules are then refined through the iterative medicinal chemistry approaches to optimize its potency, selectivity, pharmacokinetics, and safety before entering translational development. So there are three different categories of it. So we can classify them accordingly. So first one is human relevant target validation. So historically, many drug program has failed because targets validated successfully in animal models and that did not translate effectively into human disease biology. And we know that it is very important based on the evolutionary aspect. So as a result, translational research has shifted towards human-centric validation systems in order to achieve better success. So technologies such as CRISPR-based editing, patient-derived organoids, multi-omics profiling, and human tissue analysis now provide more clinically relevant biological evidence. These transition toward human relevant validation improves confidence that selected targets are biologically meaningful, therapeutically they actionable, and clinically they can be translatable as well. So next is biomarker driven drug discovery. So in modern translational medicine, biomarkers serve as critical connection between molecular mechanisms and clinical outcomes. Biomarkers enable assessment of target engagement, pharmacodynamic response, dose optimization, disease progression, and therapeutic efficacy. Simultaneously, advanced translational models, which includes in organ-on-chip system, patient-derived xenografts, and integrated PK/PD modeling, provide mechanistic insight into the therapeutic response and safety. Together, these approaches improve development precision and strengthen translational decision making. So next, biomarker and patient stratification strategy. So one of the greatest barrier to the clinical success is patient heterogeneity. So patients with the same diagnosis often exhibit profoundly different biological responses to therapy. So even the same patients can respond to the same therapy differently in different times. So biomarker driven certification addresses this challenge by identifying responder population most likely to benefit from specific treatments while minimizing unnecessary toxicity in non-responders. This approach has become particularly transformative in oncology, as well as companion diagnostics and molecular profiling now guide targeted therapeutic section and enable precision medicine strategies. So next is adoptive early signal-based design and why it is important. So conventional clinical trials are mostly rigid, so they are not flexible. So this fixed protocols comes with limited flexibility during the time of execution. So adopting these early signal-based designs can give us better or major advancement by continuously integrating emerging safety, pharmacodynamic, and biomarker data throughout the trial process. So this allows real-time modifications such as dose escalation adjustment, cohort expansion, or early termination of ineffective strategies, ultimately accelerating the development while improving patient safety and resource efficiency. So it also saves time as well and as well as money. So next is assessing the translational risk. So, despite this technological progress, approximately 90% of drug candidates still fail during the development. So, importantly, these failures are often not due to lack of innovation, but rather insufficient translation between experimental system and human biology. So, critical translational risks include poor target engagement, inaccurate disease model, weak biomarkers, unexpected toxicity, and variability in patient response. Modern translational research therefore emphasizes early risk identification through predictive models, human relevance systems, and evidence based decision frameworks. So, here we are talking about bridging the preclinical and the clinical studies and designing the first in human studies in translational context. So the transition into the first human studies represent one of the most sensitive stages of this translational development procedure. So at this point, preclinical findings must be translated into a safe and scientifically justified clinical investigation. Starting doses are determined using translational parameters that is actually followed by supported by the FDA guideline like NOFEL or MABL, while biomarkers, imaging, and PKPD modeling help in confirmation of mechanism of action and target engagement directly in humans. So increasingly adaptive phase 1 designs and seamless phase 1 to 2 transitions are improving efficiency while maintaining rigorous safety standards. So next point is about AI and big data involvement. That is our next discussion for today. So we are now entering an era of data-driven translational medicine. Artificial intelligence, machine learning, multi-omics integration, and real-world evidence are reshaping the target discovery. So toxicity prediction, patient stratification, and clinical trial optimization. So electronic health records, disease registries, and longitudinal dataset provide clinically relevant insights that complement traditional randomized trial. However, successful implementation also requires addressing critical challenges, including data heterogeneity, algorithmic bias, interpretability, and regulatory transparency. So here I'm going to talk about one of the famous story or example, that is the PCSK9 inhibitor. That is a translational success story, which suggests how the success from the wet lab can actually bridge the translational research to the clinical research. One of the most elegant example of translational drug discovery is the development of this inhibitor. So human genetic studies demonstrated that individuals carrying natural loss of function mutation in PCSK9 exhibited significantly reduced LDL cholesterol level. And we know how LDL cholesterol level is very crucial in maintaining the proper cardiovascular health, because ultimately it helps in reduction of the HDL composition in our body as well. So this PCSK9 inhibitors has different domains. We see one is pro domain, another one is V domain, another one is catalytic domain, where this threonine 61, glutamine 152, and serine 153 are the crucial amino acids playing in the catalytic role. So this provided exceptionally strong biological validation and directly accelerated the development of monoclonal antibody and siRNA-based therapies targeting this PCSK9. So importantly, LDL cholesterol functioned as a robust translational biomarker throughout the development, while clinical trials ultimately confirmed major cardiovascular benefit. So now we'll talk about the failed target case studies. So we need to learn from what has actually been failed. So it is equally important for the translational programs, which provides critical scientific insights. So analysis of unsuccessful targets often reveals limitation in disease models or biomarker selection, or it might be mechanistic assumption, or it's patient stratification strategies, because all these steps are very complicated by itself. Rather than representing isolated setbacks, these failures contribute to refinement of translational methodologies and improve future development pathways. So now we have to also consider how to manage the translational risk as well. So, to reduce this translational attrition, modern programs increasingly integrate proactive risk management throughout the development process. It is involved in all of the stages. So, human ex vivo systems, PKPD modeling, microdosing studies, and this predefined criteria help us to evaluate biological uncertainty before major clinical investment. This evidence driven framework supports more rational progression, decision, and improves overall translational efficiency. So, next, I'm going to talk about the regulatory pathway, which are actually controlling all or looking after how the things are progressing in all over the world. So, regulatory science now plays a highly integrated role in translational innovation. Agencies including the FDA, EMA, and PMDA support accelerated pathways such as fast track, prime, and breakthrough therapy designation for therapies addressing major unmet medical needs. At the same time, successful translation increasingly depends on collaborative ecosystem, which is connecting again academia, industry, CROs, all the healthcare system, patient organization, and global data sharing initiatives, which comes with different restrictions as well. So the future of translational medicine is being shaped by transformative technologies. AI-guided analytics, advanced human models, multi-omics integration, digital biomarkers, mRNA therapeutics, cell and gene therapies, and precision medicine platforms are redefining this therapeutic development. These innovations are moving translational science towards increasingly adaptive, personalized, and mechanistically driven healthcare solution. So translational case studies and strategic learning, and how can it's actually deciding the future? How is it actually deciding the future? So successful translational case studies demonstrate the value of integrating the biological understanding, biomarker strategy, human genetics, clinical evidence into a unified developmental framework. So these examples provide not only therapeutic success stories, but also strategic lessons that help in refining the translational design, reduce uncertainty, and accelerate future innovations. So to conclude this, translational drug discovery represents far more than development pipeline, and it is an evolving scientific ecosystem designed to transform biological knowledge into measurable patient benefit. So future success will depend on our ability to integrate the mechanistic biology, computational intelligence, biomarker science, and real-world evidence. So ultimately, the goal of translational medicine is not simply faster of the drug development, but smarter, safer, and more precise therapies capable of delivering meaningful impact at both individual and population levels. Thank you for your attention.
Thank you, Poshali, for this insightful lecture on translational medicine and its drug discovery aspects. So now I'm going to welcome our next presenter, Dr. Bindu Mathuri. She is a life sciences researcher with a strong interest in translational drug discovery, computational biology, and neurodegenerative diseases. Her work focuses on integrating in vitro models, computational approaches to explore disease mechanisms and thereby identify potential therapeutic strategies, particularly in cancer research. Welcome, Dr. Bindu Mathuri.
Hi to all, myself Hindu. I'm PhD graduate in biotechnology. Today we are going to have a great discussion about translation drug discovery, protein research and clinical innovation. So translation drug discovery is the process of converting basic scientific research into effective clinical therapies for patients. It bridges the gap between laboratory discoveries and medical treatment by integrating preclinical research, drug development and clinical application. It involves identifying disease targets, discovering and optimizing drug candidates, testing them in experimental models, and translating successful findings into clinical trials and patient care. The goal of this translational drug discovery is to develop a safer, more effective, and personalized therapies while reducing the time and cost of the drug development. So more, this translational drug discovery is little advanced in this area, which is called as monomer translational drug discovery. It integrates multiple disciplines such as molecular biology, pharmacology, bioinformatics, artificial intelligence, and clinical medicine. Advanced technologies including organ chip systems, 3D bioprinting, computational modeling, biomarker identification, and gene editing tools are increasingly used to improve the accuracy and success of this therapeutic therapy. So, as this important is the translation discovery, we are discussing here what are all the pits and falls of this yet to be developed and how it helps in the novel drug discovery platform. Before going into the details of the advances and all the de- um details of this translation drug discovery, I would like to give a brief about what are all this presentation is covering. It is covering conventional drug discovery approaches and its limitations, because it will be more clear if we go how this translation drug discovery is working with the basic knowledge of conventional drug discovery and its high limitations, and what are all the advanced translation drug discovery approaches and the case studies and classic examples of the uh, translation drug discovery that is being identified and are used, and what are all the future directions and conclusions. Conventional drug discovery is the traditional process of developing new medicines through sequential stages of research and testing. It mainly focuses on identifying bioactive compounds, testing them in laboratory and animal models, and later evaluating them in human clinical trials. This approach is often time consuming, expensive, and associated with high failure rates during clinical translation. Trial and error experimentation, and it depends on trial and error experimentation. Majorly, it relies on the animal models. It is a slow process, and it relies and high clinical trial failure rates are observed in the conventional drug discovery process, and it is having very limited personalization, means that it is not specific. The identified drug is not specific to any human entity or any, it doesn't involve any precision measurement factors, and it is expensive and it is time consuming, and it focuses majorly on the compound drug discovery. What One of the major bottlenecks for this conventional drug discovery and development is translation, particularly at the interface of drug discovery and early clinical development, referred to as the translational gap. This translational gap can be covered by the translation drug discovery partner. So this translational drug discovery, which is advanced, refers to the use of modern technologies and its interdisciplinary approaches to accelerate the transformation of laboratory research into effective clinical therapies. It enhances translation drug discovery by integrating advanced experimental models, computational tools, and personalized mission strategies. This facilitates the improvement of prediction of drug safety and efficacy. This approach combines several areas such as artificial intelligence, genomics, bioinformatics, organ chip systems, 3D bioprinting, gene editing, and advanced in vitro and in vivo models to better mimic human biology and disease conditions. The main aim of this translational drug discovery is obviously to reduce drug development time, lower clinical trial failures, and deliver more precise and patient-specific treatments. So, how it works to achieve all these major goals is like it uses mostly advanced human relevant models and and it proved prediction of clinical success, and it involves AI driven, as it is discussed, AI driven computation approaches, and it is having a strong biomarker integration, and this translation drug discovery is more efficient and highly targeted. It mainly focuses on the clinical applicability and patient outcomes than identifying the small molecules. So, how these advancements in clinical research, translational research have been achieved? These are achieved by several methods such as improved in vitro models and advanced in vivo models and computational tools and biomarker identification. Where if we go in detail of what are all these improved in vitro models, these in vitro models are advanced laboratory based systems designed to better mimic human tissues, organs and disease conditions outside the body. Unlike traditional 2D cultures, modern in vitro models provide more physiologically relevant environments for studying disease mechanisms, drug responses, toxicity, and therapeutic efficacy. These models include 3D structures, organoids, spheroids, organ on chip systems, and bioprinted tissues, which these models more accurately replicate human cellular interaction, tissue architecture, and microenvironment. As a result, they enhance the predictability of clinical drug testing and reduce reliance on animal experimentation. And these models are advantageous by better simulating human physiology and thus improved prediction of drug safety and efficacy, and it reduces the animal usage, and it characterizes the enhanced disease modeling and personalized medicine. So examples of these advanced in vitro models are like 3D cultures, organoids, organ on chip platforms, microfluid systems, 3D bioprinted systems. So these, as it was discussing, these improved in vitro models are having highly successful predicted in vivo outcomes, and they can be used to study multi-organ interactions, and they replicate functions of several human organs such as heart, lung and liver. So with these special benefits of these improved in vitro models, they are playing a very crucial role in the translational medicine. This table describes the certain examples of tumor organ models that so far have achieved the improvements over conventional cell culture in vitro toxicology studies. So tumor on chip, it uses human endothelial cells in microfluidic system to study tumor behavior, cancer progression and radiation effects on human microvascular. Liver on chip, it uses 3D hepatocellular cultures to study the liver specific metabolic enzymatic functions under physiological conditions. Lung on chip, it mimics lung microenvironment using alveolar epithelium and endothelial cells cultured on flexible membranes to study respiratory diseases and pulmonary drug responses. Kidney on chip, it recreates the kidney tubular structures using proximal tubule cells to evaluate nephrotoxicity and renal drug transport mechanisms. Heart on chip is one such above examples. It integrates biological mechanical signals to stimulate cardiac contractility and rhythmic heart tissue functions for cardiovascular drug testing. So likewise, lymph node on chip, bone marrow on chip are also there, where lymph node on chip uses lymph node tissues to investigate immune responses and lymphatic interactions. Bone marrow on chip is one such organ on chip model. It captures the cellular migration and hepatopoietic environment in regulator systems to study blood cell development and bone marrow pathophysiology. So these are the very classic and improved examples that supports that this in vitro organ, this tumor on chip is highly advantageous in the advanced translation drug discovery process. So then another approach that this advanced translational drug discovery uses computational models. These computational models and simulations are advanced computational computer-based approaches. They used to study biological systems, predict drug behavior, and optimize the emetic development. These methods use mathematical models, artificial intelligence, bioinformatics, and molecular simulations to analyze complex biological interactions and accelerate drug discovery. In translational drug discovery, computational tools help Researchers identify drug targets, predict molecular interactions, assess toxicity, which comes in a pharmacokinetic parameters, and simulate disease progression, which comes as a pharmacodynamic parameter. These preliminary information will facilitate the easy experimental testing. So we are all having these in hand before we are going to the experimental testing. This reduces time, cost, and failure rates associated with the traditional drug development approaches. Common computational techniques include molecular docking, molecular dynamic simulation studies, quantitative QSAR analysis, virtual screening, machine learning, and system biology modeling. These technologies enable researchers to screen thousands of compounds rapidly and identify the most promising drug candidates. However, there is no such drug that is identified or in the clinical trials that is identified using AAML. ISM 001055 is a one such drug that is come out of using AI, which is developed again as the idiopathic pulmonary fibrosis. So another approach how this advanced translational drug discovery integrates for a better outcome of a patient's comfort is that using innovative in vivo models. Innovative in vivo models are advanced living biological systems used to study disease mechanisms, drug responses, and therapeutic efficacy. These disease mechanism understanding, drug responses, and therapeutic efficacy will be studied within a whole organism. These models provide a more realistic understanding of complex physiological interactions, including metabolism, immune responses, and organ to organ communication, which cannot be fully replicated in vitro systems. Modern in vivo approaches include genetically engineered animal models, humanized mouse models, zebrafish, and alternative organisms such as C. elegans and Drosophila melanogaster. These systems are widely used in traditional drug discovery to evaluate drug safety, toxicity, pharmacokinetics, and disease progression before clinical trials. Advancements in gene editing technologies such as CRISPR-Cas9, imaging techniques, and AI-assisted analysis have significantly improved the accuracy and predictive value of these models. The key advantages of these innovative in vivo models are like better representation of whole body physiological responses, improved prediction of clinical drug efficacy and toxicity, and it supports disease modeling and therapeutic screening, enables study of complex biological interactions, facilitates translational research and precision medicine. These are commonly used in the field of cancer research, neurodegenerative diseases, and to study the drug toxicity and pharmacology and gene therapy research. So these are all the three major models that the translational drug discovery integrates for a better patient outcome. So here are some of the case studies where these different aspects of the translational drug discovery has been risen up. One such example or one such case study is that an organ on chip technology, which is developed as a lung on chip technology, it widely used to evaluate drug efficacy, toxicity, and disease progression in disorders such as asthma, chronic obstructive pulmonary disease. These systems recreate important lung functions, including airflow, tissue stretching during breathing, and cellular interactions between epithelial and immune cells. Compared to conventional cell cultures, organ chip platforms provide a more realistic representation of human lung physiology and improve the prediction of drug responses. The applications of these lung on chip technology is that testing respiratory drug efficacy, asthma and COPD mechanisms, evaluating pulmonary toxicity, modeling inflammatory lung diseases. And this facilitates the reducing the animal experimentation. The another case study is that generation of 3D bioprinted liver tissue for the drug toxicity prediction. 3D bioprinted liver tissue are advanced in vitro models designed to mimic the structural and functional properties of human liver. These tissues are created producing physiologically relevant liver-like architectures. Unlike traditional 2D cell cultures, 3D bioprinted liver models better replicate liver metabolisms, enzyme activity, and cell-to-cell interactions. This enables more accurate prediction of drug-induced liver toxicity and improves preclinical drug safety assessments. The applications of these again these 3D bioprinted liver tissues were applied in case of drug toxicity screening, hepatotoxicity studies, liver disease modeling, pharmacokinetic, and personalized medicine research. Another approach is ANML, involvement of AIML and success of AIML in the Alzheimer's drug discovery. Artificial intelligence and machine learning are revolutioning Alzheimer's disease drug discovery by enabling rapid analysis of large biological datasets and detecting potential therapeutic compounds. These computational approaches identify molecular platforms, drug targets, and candidate compounds more efficiently than traditional screening methods. AI-driven studies have identified several novel compounds capable of inhibiting the formation of amyloid beta plaques, a key hallmark of Alzheimer's disease pathology. Machine learning algorithms can also predict drug toxicity, optimize molecular structures, and support personalized therapeutic strategies. So, what is the take home message of this discussion? Translational research generally bridges the gap between laboratory discoveries and clinical applications, and how it happens. It integrates the advanced in vitro, in vivo, and computational technologies. However, it is advanced. It still has some limitations that it cannot uh, fully replicate the complexity of human physiological and immune responses, and it is having some challenges such as scalability, reproducibility, and standardization. So these are remaining the major barriers in translational drug discovery. However, the standardization, scalability, reproducibility approaches can be improved in such a way the outcome will be uniform at every aspect of testing. And advanced technologies such as organ on chip, 3D bioprinting, AI, and machine learning are transforming preclinical development. So interdisciplinary collaboration is required among researchers, clinicians, and engineers, and from the regulatory experts also for a successful translational research. Artificial intelligence is and big data analysis are enhancing predictive accuracy in drug discovery and personalization. So with all these information about translational drug discovery and how it works. Still, future translation research aims to reduce drug development costs, minimize clinical trial failures, and it should accelerate the therapeutic potential. So, this can be achieved by integrating computational biology with experimental models, and this is actually reshaping the future of the precision medicine. So I thank Anandor for presenting here to listen my talk. Thank you.
Thank you, Dr. Bindu Mantri, for this insightful lecture on translational drug discovery. Hence, we come to an end to our insightful exploration of this theme 7, translational drug discovery. Here we have successfully mastered the complex journey required to bridge the gap between laboratory research and clinical innovation. This brings us perfectly to the second session of the day, Theme 8, Bioinformatics and Computational Biology in Modern Biomedical Research. In this session, we will shift our focus to the computational tools and the algorithmic approaches required and that are currently transforming the landscape of biomedical research. Our presenters will discuss how bioinformatics is not just a support tool, rather a fundamental driver in accelerating scientific breakthroughs and enabling high throughput discovery. To lead us into this digital exploration, I would like to welcome our next group of student speakers and guest experts. Our first speaker on the lineup is Poshali Bose again. Please welcome Poshali. Poshali, are you audible?
Good afternoon, everybody. So, my name is Paushali Bose. So, today I will discuss how bioinformatics, artificial intelligence, and big data are reshaping the modern biomedical research. So, over the last decade, biomedical science has undergone.
Poshali, your screen is not visible. Can you please check it on your end? You have not started the presentation. Yes, it is visible now.
Okay. So, over the last decade, biomedical science has undergone a dramatic transformation. Technologies such as genome sequencing, electronic health records, and wearable devices are generating enormous amounts of biological and clinical data every single day. However, generating data alone is not enough. So, the real challenge lies in extracting meaningful biological insights from this vast information. So, this is where computational biology, machine learning, and AI becomes essential. So, before understanding the advanced computational tools, we must first understand why these approaches have become indispensable in biomedical research. So in this slide, I'm going to talk about three major reasons behind the rise of computational biology in medicine and life sciences. So modern biomedical research generates massive volumes of omics data. For example, a single whole genome sequencing experiment can produce nearly 200 GB of raw data. Therefore, so we know that genomic data is actually growing at this unprecedented rate, which can never be analyzed by human in a short span of time. So computational tools are no longer an optional now. So they are foundation of modern biomedical discovery. At the same time, biological systems themselves are extremely complex. Diseases like cancer, neurodegenerative disorder, they do not arise from one single mutation or pathway. Instead, they involve interconnected molecular networks operating across multiple biological levels. Traditional wet lab experiments alone cannot fully capture this system level complexity. So, computational methods help in integration of this multiomics data to understand disease mechanism holistically. The third one is the translational gap. So, this is actually a major gap between laboratory findings and clinical application. So, we know that many discoveries fail to translate into useful diagnostics or therapies. So, a large amount of data remains unexplored. So bioinformatics helps in bridging this gap by enabling this biomarker discovery, predictive modeling, which is a which is not a very old method, and precision medicine approaches. So now that we understand why computational approaches are necessary, the next question becomes how are these computational systems organized within biomedical research? So the core computational approaches include different strategies. So at this point, I have already explained why these 3 points are essential or are the core reasons for rising this computational need. So this slide introduces the overall computational framework used in modern biomedical research. So this field integrates sequence analysis, structural biology, network biology, machine learning, and statistical modeling into one interconnected pipeline that transforms raw biological data into clinically meaningful insights. So the key strategy pillars are mostly the multi-omic integration, the machine learning, network biology, and clinical translation. So basically, we know that different softwares are actually used for these purposes. So these include BLAST, GATK, Cytoscape, R, Python, TensorFlow, and PyTorch. These are very essential tools. So these demonstrate how biomedical research have become highly interdisciplinary, combining biology with statistics, computer science, and data science. So among these computational areas, one of the most fundamentals and widely used in sequence analysis and genomes, which forms the basis of many modern biomedical discoveries, is sequence analysis and genomics. Here, I'm also going to say that there are other four things that can be considered as the pillar for this core computational foundation, which not only includes multiomics integration, but also includes the clinical translation, network biology, and machine learning individually as well. So sequence analysis is one of the foundational pillars of the bioinformatics. So by analyzing the DNA and RNA sequences, researchers can easily identify the mutation, study gene deletion, study gene function, detect disease-causing variants, and understand evolutionary relationship. These applications include cancer genomics, inherited disease diagnostics, infectious disease tracking, and precision medicine. However, sequencing alone is not enough as well. Once sequencing data is generated, scientists must identify which mutations are biologically important. So, so it's not about studying the DNA and RNA sequences, but here comes the protein as well. Because at this point, we know it's not about the genomic and the proteomic data. The epitranscriptomics and epitranslational data are rising as well, where which is emphasizing on the dynamics of the DNA and RNA, and as well as the protein, to study the disease in more core level. So this brings us next to the next topic, that is called the variant calling. So variant calling is the computational process used to identify mutations by comparing patient sequencing data with a reference genome. In lung cancer, whole genome sequencing allows researchers to identify driver mutation, oncogenic variants, potential therapeutic targets, and the workflow generally includes the raw sequencing data, and then quality control, genomic alignment, and variation detection. A notation, clinical interpretation. So this is especially important in precision oncology, where therapies are selected based on the patient's individual genomic profile, but DNA sequence alone does not fully explain the disease behavior because it is lacking the dynamics of the DNA and the RNA and protein as well. So we must also understand which genes are actively expressed inside the cell. This leads us to transcriptomics and RNA sequencing. So while genomic studies states the static DNA sequence, transcriptomic studies the dynamic gene expression at the RNA level. So RNA sequencing helps determine which genes are actually active and how strongly they are expressed and how expression changes during disease progression. So application includes biomarker discovery, immune profiling, cancer subtype analysis, and single cell sequencing. So this provides a functional view of cellular activity, helping researchers understand how diseases actually behave biologically. Now that we have discussed the major biological data sets, the next challenge is understanding how researchers process and analyze these highly complex data sets computationally. I've already talked about this, but the key will still summarize the whole thing in a slide. So it summarizes the computational pipeline that is used for biological data analysis, which typically involves pre-processing the data, normalize the data, because a lot of data are actually noise. So that needs to be normalized. Alignment, statistical analysis, pathway enrichment, and visualization. As biomedical datasets become increasingly high dimensional, and traditional statistical approach alone are often insufficient. This is where machine learning and artificial intelligence become transformative. So, after discussing genomics, transcriptomics, and computational pipeline, the next major question is, how do we actually extract meaningful biological insights from these massive and complex data sets? This is where machine learning and artificial intelligence become extremely important. Machine learning allows computers to identify the hidden patterns, relationships, predictive signatures directly from biological data without explicitly programming every rule manually. In biomedical research, these methods are now widely used for disease prediction, cancer classification, biomarker discovery, drug response analysis, protein structure prediction, and personalized medicine as well. The reason AI has become so powerful in biology is because we know the data sets are complex, multidimensional, and the most complex part is its heterogeneity. which is very difficult to interpret manually. To be interpreted manually. So before understanding advanced AI applications, we first need to understand the fundamental type of machine learning used in the computational biology. So the slide introduces three major categories of machine learning used in biomedical research. So the first one is supervised learning. Second one, unsupervised, and the third one is semi-supervised learning. So each type is designed for different kinds of biological problems, depending on whether labeled data is available or not. So in biology, datasets may include mostly coming from the genomic sequences, gene expression profile, protein structure, medical imaging, or clinical patient records. So machine learning algorithms analyze these datasets to discover meaningful biological patterns and generate predictions. An important challenge in biology is that the datasets, again, it's very heterogeneous. So we have to be very rational about choosing these different types of learning procedures. So if I talk about the supervised machine learning, why it is important or when do we need to use it? So supervised learning is one of the most widely used learning approaches in the biomedical science. So in supervised learning, the algorithm learns from the label data. So the data is already annotated before. That's what is important here. So this means the correct answer or the outcome is already known to us during the process of the training. So we can say that inputs are known and corresponding outputs are known as well. But we are just feeding the machine, and because we need to train the machine using the data sets. So the primary goals are usually classification, prediction, and regression analysis. So researchers may train a model using gene expression data from patients where tumors are already classified as benign or malignant, for example. So after training, the model can predict whether a new tumor sample is cancerous based on its molecular profile. So here you see how the whole scenario is very iterative, and the more iterative it is, and the more data that we can feed into this pipeline, the better outcome it can generate to us. Applications here is actually the random forest for genomic variant interpretation can be used. So these statistical tools are very essential in this case. So random forest actually combine multiple decision trees to improve the prediction accuracy. And basically they're used commonly for identifying the genomic mutations, if the genomic mutations are even disease causing or not. Then other type is support vector machine, which usually helps in cancer subtype classification. So this is a kind of data biological set. It actually helps in categorizing the biological data set, which are useful in identifying the cancer subtype from molecular signature. For example, the markers. Neural network for drug response prediction. So neural network learn highly complex biological relationships and can predict how patients may respond to specific therapies. So in a nutshell, we can see how different statistical techniques are embedded in this supervised training network so that it can be useful in different aspects of translational drug discovery research. So supervised learning transforms historical biomedical data into the predictive clinical intelligence. That's what we can say in the summary. So now, although supervised learning is powerful, one major limitation is that obtaining this labeled biomedical data is very hard and is also very expensive, and it consumes a lot of time. So this challenge leads to another important category called semi-supervised learning. Okay. So what about semi-supervised learning? So it combines the strength of both supervised and unsupervised learning. So in biomedical research, large amount of unlabeled data are available, but labeled data is often limited because annotation requires a lot of expertise and laboratory validation as well, which comes with a significant cost. So semi-supervised learning solves this problem by training models using a small labeled dataset together with a large unlabeled dataset. This improves prediction performance while reducing the annotation burden as well. So basically, there are different ways or different methods which is incorporated in it. That is graph neural network for knowledge graph completion. So this is one of the oldest neural network where the models analyze biological interaction networks and predict missing biological relationship. Self-training models for rare disease diagnosis, which so rare diseases often lack sufficient label datasets. Semi-supervised model help improve diagnosis using this limited patient data. On the other hand, the active learning for optimal experimental selection is useful, where the model intelligently selects the most informative experiments for researchers to perform the next, reducing the experimental cost. So while supervised and unsupervised learning focus mainly on prediction, researchers also need method that is capable of discovering completely unknown biological pattern where this unsupervised learning come in play. Before going into that, I would like to mention all these neural networks that I'm talking today are widely useful, especially the biggest discovery, that is AlphaFold 3, which has been um, which has actually involved all these different types of neural network procedures. So next I'm going to talk about unsupervised learning. So unlike supervised learning, unsupervised learning works with completely unlabeled data. So this algorithm does not know the correct answers beforehand. Instead, it independently searches for hidden structures, similarities, clusters, and patterns within the dataset. So this is especially important in biology because many disease mechanisms and cellular subtypes are still unknown to us. So the goal of this major goal are actually clustering, dimensionality reduction, and the pattern discovery. So researcher may analyze gene expression profile from thousands of patients without knowing disease categories beforehand. So the algorithm may automatically discover previously unknown cancer subtypes, unique immune cell population, or novel molecular signature. So there are different techniques involved in this process. So one is TSME and UMAP, which reduce the dimension of the single cell RNA sequencing data into visual maps, helping researchers to identify distinct cellular population. Then K-means clustering is one of the popular clustering method, which actually helps in identifying the similar molecular characteristics into clusters, which includes the principal component analysis as well, which is again helps in the reduction of the dimension of the major biological variations. An important thing, unsupervised learning allow biology to reveal the pattern. This pattern identification is the crucial job of this unsupervised learning procedure that we researchers did not even know that it has been existed. So together we can say that supervised, semi-supervised, unsupervised learning form the foundation of modern AI-driven biomedical research. However, as biological data sets become even more complex, researchers increasingly rely on more advanced architectures such as deep learning, which we will discuss in next section. So after understanding the basic machine learning approaches used in biology, we now move towards a more advanced bench of AI known as deep learning. Deep learning has revolutionized the computational biology because biological systems are highly complex and contain nonlinear relationships that traditional machine learning models often fail to capture. So these deep learning models are multi-layered neural network capable of automatically learning hierarchical biological features directly from the raw data. These approaches are now transforming genomics, proteomics, all these different data sets into the proper system. One of the biggest breakthrough in this field has I have mentioned about it, that is the AlphaFold 3, where AI systems can easily predict the protein folding. But recently another software has come which can actually de- describe the protein dynamics as well, which was actually limited by AlphaFold 3. So this has dramatically accelerated the biomedical discovery because protein structure determines biological function and therapeutic targeting. So what are the different deep learning architecture methods? The first one is CNNs, that is basically used even by the even like in a normal image or photograph that has been used widely, but now it's a part of biological research as well. So these convolutional neural network are designed for analyzing spatial data like images, microscopy, histopathology, biological sequences. In this case, CNN is actually used for tumor detection, cell morphology analysis, protein secondary structure prediction, and genomic pattern recognition. So these automatically detect the important spatial features without manual feature engineering. Then comes the autoencoders, which allows the compression of the representations of highly complex biological data. They are useful for dimensionality reduction, denoising the noisy dataset, detecting the abnormalities, and input missing values in multi-omics data. So this is especially important because biological datasets are often incomplete and noisy. Then comes the transformer networks. Transformers are among the most powerful modern AI architecture. They are attention. We usually use one of the important mechanism which is called the attention mechanism. These attention mechanisms plays very crucial role in deciding which data or which input needs to be chosen or is important in comparison to the other data set available in comparison to the whole data set. So basically, it helps in understanding the attention mechanism by considering this long range interaction. So we understand that how it is important for understanding the network biology. So these applications mostly include protein language model, biomedical literature mining, drug target interaction prediction, and genome sequence modeling. So major breakthrough is again AlphaFold, that is done by DeepMind. So AlphaFold demonstrated that AI can actually predict the protein tertiary structure with remarkable accuracy, but it has limited capacity in explaining the structure of the RNA because again for its dynamics. So where RNN or recurrent neural network can be helpful to some extent. So they are useful for genomic and sequence analysis, RNA structure prediction, time series patient monitoring, and gene regulatory network modeling. These architectures demonstrate how AI is becoming increasingly capable of understanding biological complexity, but now AI systems are evolving beyond prediction models into autonomous intelligent system known as agentic AI. So agentic AI represents the next evolutionary stage of artificial intelligence. Traditional AI systems mainly analyze the data and generate predictions. However, agentic AI can reason, plan, make decisions, adopt dynamically, and autonomously perform tasks. Sounds like a man, human. In biomedical research, these systems may design experiments, analyze outcomes, generate hypotheses, optimize workflows, iteratively improve the scientific understanding. So agentic AI acts more like an intelligent research assistant, right, capable of autonomous scientific reasoning. These systems combine multiple AI capabilities, including neural language processing, computer vision, predictive analytics, and reinforcement learning. So I'm repeating the word reinforcement learning, which is very crucial. This could significantly accelerate the pace of biomedical discovery by reducing human workload and shortening research timelines. So the impact of these advanced computational systems is already visible across multiple biomedical domains. So in this slide, I'm going to talk about how AI and computational biology are transforming multiple areas of biomedical research simultaneously. The first one is genomics and transcriptomics, which comes with terabytes of data. And along with that, the terabytes of data includes a lot of noise. So deep learning models can now predict regulatory elements, splice site, enhance the promoter interaction, and disease-associated mutations. Single-cell transcriptomics also allows classification of cellular population with unprecedented resolution. Next, drug discovery and development. AI is dramatically accelerating this field. So generative models can design novel molecules, predict toxicity, estimate pharmacokinetics, and optimizing drug candidates, which usually takes time if we do it in wet lab. Virtual screening systems can computationally evaluate billions of compounds before wet lab even testing. These reduces cost, time, and experimental burden. Then protein structure and function. AI models predict protein folding, binding affinity, protein interaction, therapeutic protein design. This has transformed structure-based drug discovery. Then medical image analysis, which is the crucial part of the clinical research. So deep learning models analyze MRI, CT, histopathology, and renal imaging and echocardiography. These systems can detect disease patterns with near expert level accuracy, and this is where we can see how how incorporating the AI can be useful in identifying the patterns, which can often be ignored by the human. As these AI systems become more generalized and scalable, researchers are now developing large universal models known as foundational models. So what are these models? And so these are large-scale AI models trained on enormous and diverse data set. And we know how diverse the human population is if we consider different countries. So based on that, different molecules, even for example, hemoglobin, it is differently glycosylated based on different populations. So at this point, we understand how ethnicity is very playing a crucial role in this kind of clinical drug discovery. So instead of building separate models for every biomedical task, foundation models can learn generalized biological representations that can later be fine-tuned for specific applications. So this represents a major shift in computational biology, which includes pre-training on massive data sets, where we are actually training the model using this all sequence, protein structure, genomic data, biomedical literature, and so on. These allows this model to learn the broad biological knowledge, like you're teaching them all the aspects of biology so that it can actually interconnect all the part of the science. Then self-supervised learning, instead of relying entirely on manually labeled data, foundation models learn from unlabeled data using predictive objectives like mass token prediction, sequence reconstruction, and contextual learning. This contextual learning is a very crucial part of the graphical neural network as well. So after pre-training, the models can quickly adapt to disease prediction, drug interaction analysis, biomarker discovery, rare disease diagnosis, and as new biomedical data becomes very available, foundation models are without continuously improving without complete retraining. However, despite their transformative power, several important challenges still remain. So, although AI and computational biology offer tremendous potential, there are several critical limitations and concerns. Machine learning, that is the data quality and availability. So, machine learning models are highly dependent on data quality. Biomedical datasets often suffer from missing values, noise, batch effects, selection bias, limited diversity, poor quality. Data can produce inaccurate predictions. And then validation and reproducibility. Computer predictions must always be experimentally validated. Without rigorous validation, overfitting, false discoveries, and non-reproducible findings can definitely occur. Then comes ethical and privacy concern, which is one of the biggest concerns. Biomedical data contains highly sensitive patient's information. Researchers must ensure patient confidentiality, secure data handling, and compliance with regulation like GDPR and HIPAA. This algorithmic bias is also a major concern because underrepresented populations may receive less accurate prediction. And then comes the computational resources, which is very costly. Training large AI system require high level of GPUs, which involves a lot of energy as well. TPUs and cloud computing and high performance cluster. These resources are very expensive and inaccessible to many institutions for their maintenance. Model interpretation, many deep learning modern systems behave like a black boxes. In clinical settings, doctors and regulatory agencies need transparent explanation behind AI generated decisions. To overcome some of these limitations, researchers increasingly integrate multiple biological data sets together into unified systems. Next, we will talk about how integration is important at this point. So, multi-omic integration combines different biological layers such as genomic, transcriptomics, proteomics, metabolomics, epigenomics, and clinical data. And the eventually new field which is coming into play, that is the epitranscriptomics. This creates a more comprehensive system-level understanding of disease. Instead of analyzing one biological layer in isolation, researchers study how multiple molecular systems interact together. So ultimately, these integration help converting the raw biological data into clinical actionable decisions. This growing integration of AI, big data, and biology is one of the reasons why the field is currently a major hot topic and at a technological turning point as well. So we can say this field is at an inflection point, because we can say that biomedical research is currently experiencing a historic transformation because several major technological advances are converging simultaneously, which includes rapid decrease in sequencing, explosive growth of biological data, advances in AI, cloud computing, and improved computational infrastructure. Previously, many biomedical problems were too long or too complex to analyze effectively. Today, AI systems can process and interpret the biological information at unprecedented scale and speed. This convergence has created an inflection point, meaning the field is entering at a phase of acceleration of accelerated scientific progress and innovation. As computational systems become increasingly integrated into healthcare and biomedical decision making, ethical governance also becomes extremely important. So basically, as AI becomes more deeply integrated into biomedical research and clinical care, ethical responsibility has to be considered very well. Important clinical concerns include patient privacy, informed consent, algorithmic fairness, transparency, and responsible AI deployment. Researchers must ensure that AI systems do not reinforce healthcare inequalities or produce biased medical decisions. Another major issue is explainability. Clinicians must understand how AI systems arrive at conclusive conclusions before trusting them in real world patient care. Despite these challenges, the future direction of computational biomedicine remains extremely promising. So the future of computational biomedicine will likely involve highly intelligent, integrated, and autonomous systems capable of transforming healthcare at every level. Future systems may combine genomics, wearable devices, imaging, clinical records, and real-time patient monitoring into unified predictive healthcare platforms. AI may eventually support early disease detection, personalized therapy, automated diagnosis, and real-time treatment optimization. This could fundamentally shift medicine from reactive treatment toward predictive and preventive healthcare. Several emerging technologies are expected to drive this transformation even further. So, what are the future directions? Quantum computer is one of the famous techniques that is playing important role in physical chemistry, but we can also include it here, like quantum machine learning, where quantum computing may eventually solve highly complex molecular simulations much faster than classical computers. This could revolutionize drug discovery, protein simulation, and system biology. Again, multimodal integration, where future AI system will integrate multiple data types simultaneously, including genomics, imaging, clinical notes, and wearable sensor data together. This creates holistic patient representations. Then autonomous laboratories, where AI-controlled robotic laboratories may automatically perform experiments, analyze results, and optimize scientific workflow. I believe there is still some time to have this happen in a real world. Then precision medicine at scale, where AI-driven systems may generate highly personalized treatment strategies based on each patient's personal needs. So next is, what is the key takeaway? So we can say that advanced computational approaches are fundamentally transforming biomedical research and healthcare, and the convergence of artificial intelligence, big data analysis, and in bioinformatics, along with system biology, have created a new paradigm of data-driven biomedical discovery. So AI systems can now analyze massive biological data set, identify hidden patterns, hidden molecular patterns, I would say, accelerate drug discovery, predict disease mechanism, and support personalized medicine. However, successful implementation requires rigorous validation, ethical responsibility, interdisciplinary collaboration, and clinically meaningful interpretation. So the future of medicine will increasingly depend on collaboration between computational scientists, biologists, clinicians, and engineers. So the future of biomedical science will not only be biological anymore, so it will be definitely computational, intelligent, and deeply data driven. Thank you.
Thank you very much, Poshali, for that detailed and an excellent lecture. So let's all now take a break for 10 minutes and we'll meet again in order to continue the same theme. Yeah, we'll take a break. Hello all. Is everyone back? I'm not able to see Jansi yet. And so with we wait for 5 more minutes. Okay, so let's start the second half of the discussion today. Uh, so we are continuing with the same theme, bioinformatics and computational biology in modern medical research. So the next presentation will be mine. So I'll introduce myself here. Hi, I'm TV Venkateshwaran. I am a master's graduate from biotechnology and I have some working experience. I have worked on rare diseases, especially in clinical genomics, and my future research interests are AI in drug discovery of rare diseases. So today I have a recorded session of computational biology in precision medicine. So yeah, the presentation can start now.
So we're going to talk about computational biology in precision medicine today. So the main theme we are going to discuss today is basically we are exploring a paradigm shift in healthcare. So earlier, the diagnosis was based on the anatomical structures. Say, for example, if a particular, if there were two or three patients having retinal disease, the target used to be the eye, or if there were three or four people with lung disease, the target used to be the lung. So basically, the treatment was based on a particular symptom. But nowadays, research has shown that multiple patients can have the same symptom, but they'll be having different molecular drivers driving those disorders. So, what is the engine to find out these varied biomarkers or these varied molecular markers? So that engine is computational biology. So coming into the basic foundation of this computational biology is multi-omics, which has genomics, transcriptomics, proteomics, and metabolomics. So the thing is, the main challenge is integrating all these omics field into one layer, or we can say integrating all the data from all these fields into one layer, which is a major challenge. So that is where computational biology or AI can come into the picture. So in the next slide, we are having the explanation of all of them: genomics, transcriptomics, proteomics, and metabolomics. So basically, in order to explain in a brief manner, genomics is basically studying the blueprint or the DNA, and scientists look for any disease-causing variants or any variants across patients in the DNA. And in transcriptomics, the main aim is to look into the gene expression and which would reveal whether the blueprint or the DNA is active in real time. And in proteomics, we map the functional molecular machinery of the cell in order to understand the phenotypic execution. And in metabolomics, the small molecule fingerprints are tracked, and these reflect the current physiologic and metabolic status of the particular cell. So the main challenge here is integrating all this biological data here. So what comes next is the target validation. So if you look into the picture here, so it's showing how the CRISPR works. So CRISPR is basically a selective knockout system which works on the loss of function mechanism. So as we have seen in the earlier slides, there is a gRNA which comes and binds to the DNA, and it also brings the Cas9 enzyme. So these Cas9 enzyme creates a break in these DNA in both the prokaryotes and in the eukaryotes. So what later happens after the double-stranded break is rejoining of these sequences. So these when these sequences are rejoined, the process here is error prone, so there are multiple random homologous rejoining of these sequences. So this leads to a frame shift in DNA, and what happens here is the DNA is destroyed, or the DNA is inhibited from producing a protein from or if or if it is a case of a mutated DNA, and if there is a frame shift, the protein product will be inhibited or stopped there. So the next is therapeutic potential. There identifying the targets when modulated show maximum amelioration of the disease phenotype. So the therapeutic potential of the drug is considered high or efficient when that particular disease is ameliorated or reduced or completely inhibited. And safety benchmarking is an important process here, because once we administer the drugs, we, we should make sure that there is no particular side effect or a toxic effect or a harmful effect in that animal. So first, people, once the target is identified, validated, the hit is discovered, and after the drug forming process, after the drug is formed and ready, after all the modifications, before going into patient testing, there is a preclinical study. There are in vitro and in vivo testing done, and basically in vivo people scientists use mice models in order to study these drugs, and the effect of this drug is tested. If the is tested and if the drugs seem to be like toxic, or if it is affecting off target, and if the in vivo models like mice are dying due to these drug effects, then that drug is stopped from using, and they try to modify those drugs. So now in the next slide, we are looking into the big data explosion. So what does this graph explain? So the x-axis is the exponential growth in omics data, which requires AI for meaningful interpretation, and the y-axis shows the data size. So EB represents exabytes, PB represents petabytes, and TB represents terabytes. So these are basically large data. Uh, so what is happening here is these biological data, as per during every year, is just expanding rapidly, and the data are just accumulating. But the main problem here is interpretation. So in order to interpret these data and give a meaningful insight into this, and to give a meaningful insight of this data, it shows that AI is required. So the years from where it starts is from 2005 to 2025. So the data amount starts increasing throughout the year, and by 2025 the data size reaches to exabytes. So this indeed shows interpretation becomes a challenge, and AI is indeed needed in order to extract a meaningful interpretation of these data, of these biological data sets. So we are coming into the AI machine learning pipeline. So there are three processes. So the first process is pre-processing, then feature extraction and prediction. So what happens during pre-processing? So the data is cleaned for any noisy background data. If there are any noisy background data, uh, the data is clean, it's normalized, and the heterogeneous data are integrated from multiple sources into unified structure. So basically, heterogeneous data is, in order to explain in layman sense, we can say it's a varied data. So if I have to quote an example, like different disorders can have variable biological markers, and there can be variable disorders as per the geographic location. So all these data are integrated during the pre-processing phase into a unified structure. And then comes the feature extraction, where key biological signatures or biomarkers are identified, and they are distinguished, and these biomarkers are distinguished based on the disease states when compared to the healthy tissues. And the next process in this pipeline is prediction, uh, where deep learning models or deep learning languages are used to predict drug response and patient outcomes with very high precision. So, what are the common deep learning models that are used in this prediction pipeline? It is CNN and RNN. CNN is computational network, computational neural network, which studies the pathology images, and RNN is recurrent neural network, which looks into the genetic sequences or the genetic signatures in any disease phenotypes. So in the next slide, we are going to look into the clinical success. Uh, and this slide compares how is the clinical success across the traditional trials and the biomarker driven and other precision trials. So before going into that, so here the computational tools are used, the right patients are selected, and the signal to noise ratio are made sure they are reduced. So what shows here is the precision trials and the biomarker driven trials are much more efficient and are much more successful than these traditional trials. So that is what is given here. The data-driven trial designs yield 4x higher efficacy markers. So coming into the structural biology aspects, which is the molecular, which gives the molecular precisions. Uh, Here, scientists visualize the binding sites. So indeed, computational biology is moving across microscopes here. So if you see, there are different methods where we use to identify the structure of the protein. So one of them is X-ray crystallography. So in X-ray crystallography, protein structures are used in order to find out the structure of the protein. So through the protein crystals, there is an X-ray passed, which is then used for analyzing the structure of the protein. And then in cryo-EM, scientists used frozen data, which is then captured using which is then captured using camera. So what is captured using camera is the 2D particle in cryo-EM, and then there is a computational based structure prediction in terms of cryo-EM. So both these X-ray crystallography and cryo-EM do take into account the protein data or the protein structure. So basically, here you need the protein in hand in order to study the X-ray crystallography and cryo-EM based structures. Whereas in AlphaFold, it uses the deep learning algorithms in order to predict the 3D structure of proteins. So AlphaFold usually uses deep learning architectures, and it analyzes the various amino acid sequences adjacent in every protein, and then combines the protein into a 3D structure. In silico docking is used for testing billions of drugs or hit molecules computationally. Like if, for example, you can look into this picture where the protein is a lock, and the molecule, the ligand, or any molecule or the drug molecule can be the key for it. So once they come into the binding site, the softwares can show how these targets are getting affected by those drugs. So now we have a table. So what do these tables suggest here? They show us the different hit hit discovery method or drug discovery method. What is the mechanism that is used? How is the efficiency and how is the speed of these methodologies? So the first one, you have the high throughput screening, and these high throughput screening are automated robotic testing where the speed is moderate, and if you look into the efficiency, it is low. And the next process, you have a virtual screening. So what exactly this virtual screening is? You use the in silico molecular docking, which we tested before, and the speed is comparatively faster, and even the efficiency is higher in this method. And then you have a fragment based testing where you have different weaker molecules which are then combined to create a higher potent molecule, which are then combined into a higher potent molecular drugs. So that's why the mechanism here is building the lead molecules. The speed is low and the efficiency is higher here. And in natural products, you do extract from plants or fungi, you do extract compounds from all these natural products, and the speed depends on what kind of products you use here, and the efficiency is higher. So basically, the table compares different drug discovery methods, and it delves into the efficiency of those methods. So, what do we have in the modern therapeutic modalities? We have various drugs, various types of drugs here. So some of them are monoclonal antibodies like trastuzumab, where they bind the receptors and then they start inducing at the molecular level. So once they bind onto the receptors, onto the surface receptors, and receptors, they start producing immunological process. So this is very useful in terms of immunodeficiencies. And then you have gene editing, especially the CRISPR-Cas9 molecular scissors. This is the molecular scissors which we have looked into before, where the DNA fragment is cut into, and then It works on the basis of loss of function. There once the double when there once if there is any double-stranded break, the DNA starts repairing based on an error-prone process. There is a rejoining process where there multiple where many nucleotides would be added randomly, which can cause frame shift mutation, and therefore uh, if there are any abnormal protein synthesized, those will be blocked. So this CRISPR-Cas9 molecular scissor is a particular knockout process. And you also have gene therapy where you give the blueprint in order to replace the defective DNA or the defective gene there. And also you have RNA therapeutics or siRNA therapeutics. So these actually fine-tune the gene expression at the transcript level. When you take RNA therapeutics, they will directly synthesize the protein inside the cell, and siRNA can be used to reduce the function, or it is also called gene silencing. So, what is this integrated ecosystem here? So, this is a potential future in terms of the healthcare process. So you have certain foundational technologies like wearable sensors where you have all those smart watches which can actually detect your heart rate or any other health factors that are inside your body, and that those uses AI algorithms. Then you have energy efficient computing and flexible and compatible, and they have flexible and compatible materials. And then they can be applications in healthcare. So as we have spoken here about these wearable sensors, it is for health monitoring and even for personalized treatment. So what all this does is like once if we apply all these AI driven processes in healthcare, you can create a digital twin for every patient. So how you can look at digital twin is it's a computational copy of a particular patient. So before testing in that patient, you can test those drugs or any other therapies in those digital twin in order to see how those therapies react in that twin. And then once if those are successful, you can move into the actual patient. So this is how personal care can get shaped in the future. So thank you very much, and I'm open to any questions.
So, and then, so next in line, we have Miss Pompala Jansi. She is currently a final year student in BE Bioinformatics at SciMaths Engineering College. And she's going to take a lecture on the AlphaFold revolution. Over to you, Jansi.
Hello everyone, my name is Jansi, and as a part of this lecture series, today I'm going to present on the AlphaFold revolution, how AI is transforming protein structure prediction and drug discovery. The presentation you are looking at is called the AlphaFold revolution, and the word revolution is not an exaggeration. Here we are going to talk about a technology that has fundamentally changed how we understand the life at molecular level and how we discover the drugs to treat diseases. Before I get into the science, I want to ask you something. Think about the last time when you took a painkiller, an antibiotic, or any prescribed medicine. Every single one of those drugs were designed to fit into a specific pocket in a protein, like a key fitting a lock. To design that key, scientists first had to know the shape of the lock. And figuring out the shape of proteins, that was the hardest part. That was the 50-year problem until AlphaFold. So today across these 11 slides, we are going to cover the protein folding problem and why it was so hard, how AlphaFold works, the stunning 2020 breakthrough, and what all of this means for drug discovery, global science, and the future of medicine. Let's begin. To understand why AlphaFold is so important, we first need to understand the problem it solved. And the problem lives at the heart of molecular biology. Every living cell is a factory, and the workers in the factory are proteins. Your muscles contract because of proteins. Your immune system fights diseases because of proteins. Your DNA is read, copied, and repaired by proteins. Enzymes that digest your food are proteins. Receptors on your neurons that allows you to feel joy, pain, hunger are proteins. They are, in a very literal sense, the machinery of life. Now, here is the key insight. A protein's shape determines its function. A protein is not just a string of chemical building blocks. It is precisely folded three-dimensional structure, and that shape is everything. Changing the shape even slightly and the protein stops working. This is what happens in diseases like Alzheimer's, Parkinson's, and cystic fibrosis, where the proteins misfold and the consequences are devastating. The problem, figuring out the shape, was extraordinarily difficult. The traditional experimental methods like X ray crystallography, cryo-electron microscopy, and NMR spectroscopy are powerful, but they require months to years of work per protein. You must grow protein crystals, fire X rays at them, and use complex mathematics to reconstruct the 3D shape from the scattered data. This process is expensive, sometimes costing hundreds of thousands of dollars per structure, and it fails entirely for many proteins that simply refuse to crystallize. By 2020, after decades of effort, the global scientific community had experimentally determined around 170,000 of protein structures. That sounds like a lot, but until you realize there are over 200 million known proteins in nature, we have solved less than 0.1% of the problem. And this gap has a name. In 1969, a physicist named Cyrus Leventhal pointed out what became known as Leventhal's paradox. If a protein randomly tries every possible configuration to find its final folded shape, it would take longer than the age of the universe. Yet in a living cell, protein folds in a microsecond. Nature has solved this problem, but biology could not tell us how. That is the puzzle that AlphaFold came to solve. The AlphaFold story unfolds across just 6 years. 6 years from the promising first attempt to a Nobel Prize. Let me walk you through the timeline because each milestone matters. In 2018, DeepMind, the London based AI research laboratory, which was owned by Google's parent company, Alphabet, has entered a biennial competition called CASP-P. CASP-P stands for Critical Assessment of Protein Structure Prediction, and it is essentially the Olympics of protein folding. Here, the teams from worldwide submitted their best models of unknown protein structures, and the predictions are compared against experimentally determined answers. AlphaFold 1 entered CASP 13 and won, not by a little, but by a margin that made the entire structural biology community sit up and take notice. But that was just the beginning act. 2 years later, at CASP 14 in 2020, AlphaFold 2 did something that no one had ever seen before. It predicted protein structures with accuracy that is indistinguishable from experimental methods. The median backbone error was less than 1 angstrom, that is 1/10 billion of a meter. For many proteins, AlphaFold's predictions were as accurate as the experimentally determined structure. The scientific community described it as a watershed moment. Some called it biology's moon landing. By 2021 and 2022, DeepMind partnered with EMBL's European Bioinformatics Institute to launch the AlphaFold protein structure database. Initially containing around 350,000 structures, it rapidly expanded to cover over 200 million proteins, essentially the entire known proteome of life on Earth. And it was completely free. Any scientist anywhere in the world could access it with the web browser. In 2024, AlphaFold 3 was released, taking the technology beyond proteins to predict the structure of DNA, RNA, small molecule ligands, and full molecular complexes. That same year, and this tells you everything about the significance of this work. Demis Habbison and John Jumper were awarded the Nobel Prize in Chemistry. The Nobel Committee cited the transformative impact of AlphaFold on our understanding of the machinery of life. 6 years, contest winner to Nobel Prize winners. This is the speed at which AI is reshaping the science. Now, let's look at how AlphaFold 2 actually works. I want to emphasize this is a high level overview. The full technical details spans a 40-page nature paper. But the core architecture is beautiful in its logic, and I think you will appreciate it even without a deep machine learning background. The starting point, step 1, is simply the amino acid sequence. Every protein in our DNA is encoded as a chain of amino acids, and that chain can be read from a gene database. This is the raw input. You are essentially giving the model a strike of letters, each representing one of the 20 amino acids, and asking it to predict how that string folds into a 3D object. Step 2 is multiple sequence alignment or MSA. This is where AlphaFold becomes clever about evolution. Over billions of years, proteins have evolved. If two amino acids in a protein tend to mutate together across different species, what biologists call as coevolution, it is a strong signal that those two residues are physically close to each other in the folded structure. AlphaFold gathers thousands of related protein sequences from across the tree of life and mines them for exactly these coevolutionary signals. And it is essentially reading the fossil record of protein evolution to infer present day structure. Step 3 is evoformer, which is a AlphaFold's central innovation. This is a stack of 48 deep learning layers, each of which is a transformer, the same architecture behind the large language models like GPT. But instead of processing words, the evoformer processes two representations simultaneously, the sequence representation and a pair representation that captures the relationship between every pair of amino acid positions. These two representations communicate with each other across the 48 layers, progressively refining the model's understanding of how the protein is folded. Think of it this way for an easy understanding. The evoformer is learning the grammar of protein folding from evolutionary data. The same way a language model learns the grammar of English from the text. Step 4 is structure module. Using the refined representations from the evoformer, the structural module iteratively builds the three-dimensional atomic coordinates of the protein. It does this using a process called invariant point attention, which ensures that predictions are independent of how you orient the protein in space, which is a crucial property for physical correctness. And step 5 is the output. Three-dimensional atomic coordinates for every atom in the protein are accompanied by a confidence score called PLDDT, which is per residue local distance difference test. This score, ranging from 0 to 100, tells you how confident the model is at each position. A score above 90 means the prediction is likely to be very close to the experimental structure. And below 50, the model is essentially saying it doesn't know, which is itself using the information about disordered regions. The four key innovations at the bottom of this slide are worth highlighting. The end to end training directly on experimental structures, the dual sequence and pair attention mechanism in the evoformer, the per residue confidence scoring, and the use of structural templates. Together, these makes AlphaFold 2 not just accurate but interpretable. You know where to trust the predictions and where to be cautious. The bar chart on this slide tells the story of CASP14 more powerfully than any words can. Look at it. Every other team in the competition, including the team of experts scientists, using the best methods available, scored between 58 and 73 on the GDT scale. GDT stands for Global Distance Test and it measures how closely a predicted structure is to be the experimental answer. A score of 100 would be a perfect prediction, and a score of 70 was considered excellent before 2020. AlphaFold 2 scored 92.4. The number was so far beyond the competition that CASP organizers initially wondered if there had been some error. There had not. For many of the test proteins, AlphaFold 2's prediction were essentially indistinguishable from the experimentally determined structures. The median backbone root mean square deviation, which calculates the average displacement of predicted atoms from their true positions, was just 0.96 angstroms. To put that in context, individual atoms are about 1 to 2 angstroms in size. Here, AlphaFold was predicting protein structures with near atomic accuracy from sequence alone in seconds. What made this result particularly startling was the range of proteins it could handle. Previous methods tend to work well on proteins that are similar to ones already exist in the database, and they fail on genuinely novel folds. Here, AlphaFold 2 maintained its accuracy even on the proteins with no close structure relatives. The proteins that had never been solved and whose folds were completely unknown. These were the proteins that had stumped experimental methods for decades. John Moult, one of the founders of Caspi, said at the time that this was a stunning advance that could change the field forever. Andre Lupus, a structural biologist who had been trying to solve one particular protein structure for a decade, described receiving AlphaFold's prediction and realizing it was correct within minutes. A decade of experimental work solved by an AI in seconds. A watershed memory for biology is equivalent to landing on the moon. That quote captures the scientific community's reaction. This was not incremental progress, this was a phase transition. Let me put the impact of AlphaFold in the most concrete terms possible, before and after. This table summarizes five dimensions of structural biology, and across every single one, the change is not incremental, it is transformational. Firstly, time per structure. Before AlphaFold, solving a protein structure experimentally took months to years, and you had to purify the protein, grow crystals, which often failed, and spend months doing computational analysis. And after AlphaFold, type in a sequence and get a structure. It hardly takes seconds to minutes. Second, cost. Experimental structure determination costs anywhere from $100,000 to over $1 million per protein. And here the AlphaFold is free, completely, openly, freely accessible. Any student with a laptop and an internet connection can predict a protein structure today that would have cost a grand cycle a decade ago. Third, accuracy. Now, I want to be careful here. Experimental structures are still the gold standards, but for the vast majority of proteins, AlphaFold's predictions match the experimental structures so closely that they are suitable for downstream applications like drug discovery, functional annotations, and evolutionary analysis. Fourth, scale. Before AlphaFold, the PDB protein data bank contained around 170,000 experimentally determined structures. And the AlphaFold database now contains over 200 million predicted structures. This is an increase of more than 1,000-fold in the available structural knowledge of life. And lastly, the democratization of structural biology is not a side effect, it is a revolution. Researchers in low income countries, students at institutions without electron microscope, independent scientists that everyone now has access to the same structural information that previously required decades of expensive infrastructure to generate. If AlphaFold 2 was biology's moon landing, then AlphaFold 3 is the mission to Mars. It does not just predict proteins, it predicts the full biologist context in which protein operates. Next, AlphaFold 3, the next generation. Here is the thing about biology, proteins rarely work alone. In a living cell, proteins interact with the DNA, with RNA, with small molecule metabolites and drugs, with metal ions, and with each other in the massive complexes. A drug does not just bind to a protein in isolation, it binds to a protein that is already interacting with the DNA or wrapped around another protein or sitting inside a membrane. To truly understand and engineer these interactions, you need to model the entire molecular environment. AlphaFold 3 does exactly the same. On the left of the slide, you can see the six molecular types, and it can now handle them. That are proteins, DNA, RNA, small molecule ligands, metal ions, and full molecular complexes combining all of the above. This architectural change that made this possible is a shift from recycling based approach of AlphaFold 2 to a diffusion based architecture. Diffusion models, the same class of AI models that generates photorealistic images from text descriptions, starts with pure noise and progressively refine it into a coherent output. In AlphaFold 3, this means starting with a cloud of random atomic coordinates and iteratively refining them towards the true structure. This approach is more flexible and generates more naturally to different molecular types. The numbers are striking. On protein ligand interfaces, the precise binding geometry that determines whether a drug will work, where AlphaFold 3 reduces the prediction error by 50% compared to AlphaFold 2. This is not a minor improvement. In drug discovery, the difference between a prediction that is 50% more accurate can be the difference between identifying a drug candidate and missing it entirely. AlphaFold 3 is not a better version of AlphaFold 2, it is a fundamentally more complete model of molecular biology. For signal transduction research, for understanding gene regulation, for designing drugs that target protein DNA or protein RNA interactions, conditions like cancer, viral infections, and genetic disorders, AlphaFold 3 opens entirely new scientific territory. Let me now connect AlphaFold to drug discovery. The table on this slide shows the traditional drug development pipeline across the AI powered equivalent. The first step in drug discovery is target identification, finding the right protein to go after the particular disease. Traditionally, this requires years of biological research, knockout experiments, genetic association studies, and protein interaction mapping. With AlphaFold providing instant structural information, target identification can now be done in weeks. Second, structural solving, which once took 1 to 2 years per target, now takes minutes. This single change has the potential to collapse a decade of the front end of the drug discovery pipeline. Third, virtual screening. Computationally, docking thousands of candidate molecules into a protein's binding site to find the best fits has also been transformed. The recent wins column on the right gives you a sense of where this is already making a real difference. AlphaFold structures have been used to identify vaccine antigens against malaria. New drug candidates against antibiotic resistant bacteria have been discovered using AlphaFold guided virtual screening. Cancer's neogen therapy, designing personalized treatments based on a patient's unique tumor protein variants, has been accelerated using AlphaFold predictions. Chagas disease, a neglected tropical disease that affects millions of people in Latin America, now has promising drug leads identified through AlphaFold based research. These are not hypothetical future applications. These are active programs running in pharmaceutical companies and academic labs right now. I want to be honest about one thing. Clinical trials still take 5 to 10 years. Regulatory approval, patient safety, long term efficacy, where AI cannot shortcut this and it should not. The value of AlphaFold is in the preclinical phases, like from getting idea to drug candidate faster, cheaper, and more accurately. The goal is to put more good candidates into the pipeline so most of them survive to become the medicines. The global impact of AlphaFold 3. 200 million protein structures that are publicly available, and 190 countries accessing the database, 2 million researchers using AlphaFold tools, thousands of papers being published citing AlphaFold every year. This is not the impact of a tool that only elite laboratories can use. This is genuinely global science. And the six application areas highlighted in the grid below represents the breadth of what this means for humanity. Precision medicine. AlphaFold allows researchers to immediately predict how a disease causing mutation changes a protein structure and therefore understanding why it causes disease and how to fix it. This is the foundation of personalized medicine. Next, vaccine development. To design a vaccine, you need to present the immune system with an antigen, a piece of pathogen that it can learn to recognize. By knowing the structure of this antigen, it allows the immunologists to engineer it for maximum immune response. And AlphaFold has already accelerated antigen design for malaria, RSV, and various bacterial infections. Next is enzyme engineering. Enzymes are nature's molecular machines, and we use them in everything from food production to pharmaceuticals to industrial chemistry. AlphaFold has been used to design plastic degrading enzymes like PETAs, potentially helping to address the global plastics crisis. It is being used to engineer the enzymes for more sustainable chemical manufacturing. Next is synthetic biology, the most forward looking application. Designing entirely new proteins that do not exist in nature, that are built from the ground up to perform specific functions. Here, AlphaFold combined with generative tools like AI tools, RF diffusion, and protein MPNN is making this a practical reality. We are no longer just reading the proteins that evolution gave us. We are beginning to write new ones. And I want to end the slide by emphasizing antibiotic discovery. Antibiotic resistance is one of the gravest public health threats of the 21st century. The WHO, World Health Organization, estimates that by 2050, drug resistant infections could kill 10 million people per year. AlphaFold guided research is opening up new bacterial targets, proteins that antibiotics have never hit before, and that could become the next generation of antimicrobials. No technology, however transformative, is without limitations, and I think it is important to be honest about what AlphaFold cannot do. Good science requires both excitement about possibilities and clarity about boundaries. The most significant limitation is dynamics. Proteins are not static objects. They move, they flex, they undergo conformational changes as they bind to partners. AlphaFold predicts a static structure, the ground state, but not the range of shapes a protein can adopt. For many drug targets, the dynamics matter enormously because drugs often work by locking a protein in a particular conformation rather than simply occupying a binding site. Next, membrane protein remains challenging. These are the proteins embedded in cell membranes like ion channels, G protein coupled receptors, transporters, etc., and they account for the target of roughly 50% of all the drugs. Their hydrophobic nature and dependence on the lipid environment make them harder to predict the accuracy. And critically, experimental validation remains essential. AlphaFold is a prediction tool, not an oracle. For high stakes applications, clinical drug candidates, for instance, you will still need experimental confirmations of the predicted structure. This is not a weakness, it is how science should work. Now, the future, and this is genuinely exciting. The next frontier is generative protein design. Tools like RF diffusion and ESM fold are using their structural knowledge encoded in AlphaFold to design the proteins that do not exist in nature. Proteins that are engineered to perform the specific functions from drug discovery to biosensing to target cancer therapy. This is the synthetic biology at the molecular level. Full AI drug development pipeline are becoming a reality. Companies like Isomorphic Labs, DeepMind's spinout are interrogating AlphaFold with molecular dynamic simulations, ADMET predictions, and clinical data to build end-to-end pipelines from target identification to drug candidate in months rather than years. And integrating with single cell genomics and proteomics will allow researchers to connect protein structure to the cellular context and understanding not just what a protein look like but how its shape changes in different cell types, disease states, or in response to the treatment. In conclusion, AlphaFold has ushered in a new era of AI powered biology, dramatically accelerating biomedical research and life saving discoveries. I believe that sentence with every fiber of scientific convictions I have. This is not hyperbole. This is what is happening right now in the laboratories around the world. These are the references of the key papers behind everything we have covered today. And I particularly recommend the 2021 paper, the first reference, where you can find the technical details of AlphaFold 2, which I have mentioned in the second slide. And thank you so much for your time and your attention. I genuinely loved this topic and I'm happy to go deeper on any aspect of it, like the technical architecture, the biological applications, or the ethical considerations, or even the future directions of this field, particularly. Now, the floor is open to questions, and I'm happy to answer any of them.
Thank you very much, Jansi, for a wonderful and a very explained lecture. So next in line we have Sohit. So Sohit is an undergraduate researcher specializing in AI and drug discovery at the moment, and is currently working at Chang Zhangjiang University. Apologize for any wrong pronunciation here, which is at the university is in Taiwan, and he's working as a research assistant, and he would like to present one of his projects that's been recently published. Over to you, Sohit.
Hi everyone, this is Sohit. I'm very excited to be a part of the student webinar lectures day 4 again. So today I would like to talk about my very own project which is based on Alzheimer's disease, where we have done machine learning driven QSAR studies and also docking pipeline in order to identify amyloid beta A4 inhibitors in Alzheimer's disease. So without getting it, let's get started. So in this lecture, we'll discuss about what is Alzheimer's and how the drug discovery is being done currently, and the workflow of our QSAR modeling and molecular docking in order to investigate the amyloid beta A4 protein inhibitor activity, and then we'll conclude with the results and discussion. All right. So basically, what is Alzheimer's disease? So Alzheimer's disease is a chronic neurodegenerative disease that leads to gradual loss of neuronal cells, and it is the cause of 60 to 70% of cases of dementia. And pathologically, it is determined by two hallmark features. The first one is extracellular amyloid beta, which is otherwise called amyloid beta plaques, and intracellular neurofibrillary triangles, which are composed of tau protein. And APP, which is the amyloid precursor protein, actually is a transmembrane glycoprotein that is predominantly expressed in the brain. So it undergoes proteolytic cleavage via two pathways. One is non-amyloidogenic and one is amyloidogenic. So what happens in Alzheimer's disease is that this amyloid precursor protein is cleaved by beta secretase, otherwise called BACE1, and gamma secretase. This is what I've discussed in my day 2 lecture as well. So this abnormal cleavage results in the generation of amyloid beta peptides, particularly amyloid beta 42. And this leads to more prone to aggregation, which leads to plaque formation. So these amyloid beta A4 proteins aggregates in order to form the core of the senile plaques. So if we target this amyloid beta A4 protein, then we can possibly inhibit the plaque formation and also enhance clearance of them. So preventing amyloid beta aggregation is a key strategy for this disease modification, Alzheimer's disease. So amyloid beta targeted therapies may slow or halt the progression of cognitive decline. So accurately predicting the inhibitors which could go and bind to amyloid beta A4 target site is a very complex task because of the diverse nature of the protein and their interaction with the other inhibitors. So to overcome this complexity, what we did was we integrated machine learning techniques into QSAR, which is quantitative structure activity relationship, which has emerged as a valuable tool since 2000s. We have tried to integrate machine learning in this. So what we did was we took the data from the Campbell database, which is based on bioactivity data set of amyloid beta A4 inhibitor. So in the initial data set, we had like 8,900 approximate bioactivity values for around 1,498 compounds. And then we narrowed down to the compounds which were having only IC50 values. What is IC50 value? It's basically like inhibition concentration at 50%. So we wanted to take that value, and then the selected data set came around 1256. So we had a drop down out of around 200 to 300 compounds. And then from that narrowed down data set, we removed the compounds with redundant or missing bioactivity values, which is a part of data cleaning and pre-processing. And then we narrowed down to 1248 compounds. So in that compounds, we segregated them into active, inactive, and intermediate. So how did we do that? So we converted those IC50 values into logarithmic IC50 values so that they come into a range of 1 to 10. So 1 to 4 is basically inactive, 5 is intermediate, and anything above 5 is active. So based on that, we labeled the compounds, and then it resulted in a pre-processed data set. It had Campbell ID, it had SMILES, it had this bioactivity class and the standard IC50 values. And then what we did was we did data processing, which is where the core of the model comes in. So we calculated first Lipinski descriptors, which is basically rule of 5, molecular weight, uh, number of hydrogen donors, acceptors, and log p value. So we have to consider these, and then we plotted some graphs, which I'll show you next. And then we converted the IC50 values into PIC50 values, which I've said. And then we removed the intermediate class for better efficiency. So we wanted either inactive or active. And then we got a process data set, which is around 1241 compounds. And then we applied an algorithm called Kennardson algorithm in order to select the diverse compounds from the process data set in order to test it. So we took around 30 diverse compounds and we tested with our web application, which I've built. So how did we build that web application? And the model is that first we performed the EDA, which is very important, where we did basic box plots and then we did basic scatter plots and frequency plots. And then the very important thing is we calculated fingerprint descriptors for the EA83 inhibitors. And then what we did was we did feature selection where we removed the collinear descriptors and then we split the data. Training set was around 80% and test set was around 20%. And then we built a QSR model based on the training data set. If it had performed really well, uh, so we basically what we did was we trained multiple models like 42 plus machine learning models using lazy predict. Out of that we took the best model and then we took out the backend model and then put in the background of the web application. Here you can see on the right hand side where you can just give the input molecule in the form of smile and the background ML model, which is the best model which you predicted. It will give you the predicted IC50 value and it simplifies the whole process basically. So as I've said, we have performed EDA, which is exploratory data analysis. Here you can see when we plotted the graph between log P and molecular weight, you could see that there is a dense clustering of inhibitors and the molecular weight range of 200 to 600 Dalton. So 200 to 600 Dalton, you could see the dense clustering and also there is a log P value range between 2 and 7. 2 and 7, you can see a dense clustering. So we wanted to get an idea where our inhibitors chemical space was lying around. And then we also took the Lipinski rule into consideration. We wanted to know how many of our compounds are actually satisfying the Lipinski rule of 5. Here you can see that between active and inactive there is a different distribution. We have to reject the null hypothesis because here you can see in inactive we had around it is lying anywhere between 2.5 to 5 and then inactive anywhere from 2.6 to 4.8 roughly. So it had a totally different distribution. You can see the outliers as well. Uh, and then we also did number of hydrogen bond acceptors. It also had a different distribution. And then we also did number of hydrogen donors. It also had a different distribution. And then we also did for molecular weight. It also had a different distribution. So why did do this is because we wanted to have a knowledge of how a chemical space of the model which we are training for, like is it really considering the Lipinski rule of phi, is it satisfying them or not? Because Lipinski rule of phi is established by I believe the Pfizer company which has released these rules like molecular weight should be less than 500 and donors acceptor should be of certain number and every inhibitor out here has followed that rule and there are some outliers here and there but most of the compounds have followed the Lipinski rule of 5 and then this is for PIC50 value. It also had a different distribution and then as I said with the fingerprint calculation like around 880 pumpkin fingerprints and also we tried to do max and other fingerprint descriptors but due to the compute constraint we didn't do that. We just did the 881 pumpkin fingerprints and then we did an evaluation of around 40 plus machine learning models. Here you can see the graph. So in R square, so basically in order to consider a good model, you need to have an high R square, lower RMSE and less computation time. Here you can see the histogram gradient boosting regressor has a very high R square and lower RMSE. Here you can see the first one is histogram gradient boosting regressor, and the very least RMSE is histogram gradient boosting regressor, and the time taken is comparatively more, but it's still reasonable because considering like only 1 second overall. So we still consider that we gave it a less weight, but still, yeah, 1 second is a decent time. But there are other models which could give you the result in like 0.1 seconds, 0.2 seconds, but they had like poor R square or very high RMSE, which is why we didn't consider that. We took the histogram gradient Gaussian regressor and put as the backend model, and then we wanted to see what is the experimental versus predicted scatter plot look like. Here you can see most of them are aligning along the diagonal, which means you can see that experimental and predicted are almost along the same way because we had higher R square as you have seen already and also lower RMSE, and we wanted to visualize of that specific model, which is why we plotted the scatter plot. And then we wanted to know on based on what features basically it is predicting a very good prediction based on what basis. So we don't want to leave it as a black box, so which is why we plotted this. So we wanted to plot mechanistic feature importance of like 880 fingerprints which we have done. So we have done it using permutation index. Here you can see PubChem fingerprint 23, 405, 577, 697, 438, and 615. These are the few PubChem fingerprints that played a really crucial role in order to give a correct prediction for our inhibitors. So what this fingerprints refers to is given on the right hand side, which gives a description. So we gather this PubChem fingerprint descriptors from parallel descriptor source, so anyone can compute the fingerprint descriptors very easily using that tool. And then, as I've said, we have done Kennard's Spoon algorithm from the process data set. We have taken around 30 inhibitors in order to consider diverse data set. So what I mean by diverse data set is that it should have a different log-P and a different MW. So we want to have a diverse data set in order to test our model, and then that's why we have done this. So here you can see the chemical space was all lying around 227 and 200 to 600, as I said already before. And then we took a diverse data set, some from 1400 space, some from 1200 space, and some from 600 space. So we have done like the red circles represent the diversity data sets, whereas the gray circles represent the active inhibitors. And then we took those diverse data sets and we have performed molecular docking as well. So these are those compounds. These are the representative diverse compounds that were taken using Translational algorithm, and these are the structures of those. And then we have deployed a prototype using neuroic50.streamlit.app. Even you can now log in and check this website. You can just go upload your file here, and then you'll be getting those values. And we also tested with the molecular docking. We wanted to see if it's actually forming any bonds. So the first compound is actually showing certain hydrogen bonds, which is really good. And the second compound and also the third compound. So we have done molecular docking for all the three top representative compounds. You could also do for the other top five, but due to the time constraint we couldn't do that. So if I were to conclude, so we have used a total of 881, but if anyone wants to continue this work, they could use other fingerprint descriptors like max and minimo, and then here random forest and also HGBR, which is histogram gradient boosting regressor. It has performed really well, and the permutation index, as you have seen, PubChem fingerprint 23, 405, 577, 697, 438. They played a crucial role in order to inhibit the amyloid beta A4. And then our molecular docking results also supported the findings because all the diverse compounds exhibited binding energies greater than minus 6 kilocal per mole, which basically indicates that it has a stronger preference for interaction with the hydrophobic pocket of amyloid beta A4. And basically, why did we do this study? Is because if we wanted to perform a study that could offer a basic guideline or to design novel amyloid beta A4 inhibitors or modulators out there. So thank you guys. If you have any questions, you can reach out to me on soy.bioinformatics@gmail.com. Thank you so much.
Thank you very much, Shobit, for the wonderful presentation of your work. So next up is one of my presentations from the previous theme, which is drug discovery fundamentals from biological targets to therapeutic designs. So I'll be presenting about rare disease drug discovery. So the presentation can start now.
So good morning all. So today we are talking about rare disease drug discovery, which comes under the theme of drug discovery, and we are going to understand the part from biology to therapy. So the first slide is coming to the first slide. We are going to talk about what are rare diseases. So as the name suggests, these disorders are very rare in nature. So throughout the world, you have a very limited population, very limited sets of individuals who are associated with these disorders. So approximately there are 7,000 rare diseases which are known, and most of them are genetic in origin. So these disorders primarily affect neonates or infants or childhood of any children, and a lot of organs are affected in these kind of rare diseases. Some of them being the nervous system, some being the eye, some being the heart, some being there are cholestasis disorders, which are hepatic disorders as well. So the prime examples of these rare diseases are Duchenne muscular dystrophy, which is an X-linked disorder, and mutations, null mutations in the DMD gene or exon deletions in the DMD gene affect causes these kind of muscular dystrophy. And the next is Huntington disease. Then comes spinal muscular atrophy, which is primarily caused by exon 7 deletion of SMN1 gene, and Rett syndrome, which is also an X-linked disorder, which affects these disorder. So Rett syndrome is caused by mutations or variations in the MECP2 gene. Uh, so some of them mentioned here are X-linked disorders, and there are autosomal disorders such as epileptic encephalopathy. They can be autosomal dominant or autosomal recessive, and the biological causes are different for different type of epileptic encephalopathy. So moving on, so why are these rare diseases challenging? So let's come to the diagnostic odyssey timeline of these rare diseases. So the main point is, the first point is the delayed diagnosis. So why are the diagnosis delayed? So this is because number one, patients can go from one clinic to other, shifting from clinicians. So the history remains unclear here in these cases. And even if clinicians give samples for sequencing in order to identify what is the cause of these rare diseases, sometimes patients end up getting uncertain significant variants, which means the diagnosis is uncertain. So this is why the diagnosis in these cases are delayed. And coming to the next point is, as we had spoken in the first slide, it's the limited patient populations. which means it is difficult to gather data across the world and it's geographically fragmented. And the next challenge is incomplete biological understanding. So here, in most of these rare diseases, what kind of mechanism causes these rare diseases are not known. In some of them, to be specific, in some of these disorders, the mechanism of these diseases are not known. In some of them, like for example, you can say in muscular dystrophy, which we spoke in the first slide, the null variants are known to cause those diseases, but we don't know how the missense variants affects those diseases. So that is one of the challenge. And the next challenge is lack of funding, which is one of the most important challenges because the rare disease researchers R&D are underfunded compared to other blockbuster drugs like cancer. And the last challenge is fewer approved therapies. That's because the approval of these therapies are long in process. The approval process is very lengthy, and there are due to which there are very less effective treatments which are available. So coming to the drug discovery pipeline, so what goes in here? So the first point of this pipeline is the disease biology because this point is very critical before going into identifying a lead, validating it, going to identify a hit molecule, etc. So first you have to understand what is the process, I mean like what mechanism is causing these rare disorders. And once you identify the mechanism or the characteristic of these rare disease, then scientists go into the therapeutic target, where a particular molecule or a ligand is identified which causes the particular diseases, which causes a particular rare disease. We'll come into detail later. And then once you identify a target, you need to validate that target because scientists need to check if that particular target is actually causing the disease. And once the target is validated, scientists go into hit discovery because because why hit discovery is important because the thing is the hit is necessary in order to target the therapeutic target here, which means these molecules are required to interact with these targets so that the rare diseases can be reduced. And then comes the lead optimization, where they modify the chemical property and then which in turn leads to a higher potency, selectivity, and safety of these compounds or molecules. Then after all the optimization process comes the preclinical studies. So preclinical studies is an important barrier here before going into the clinical trials. So what happens in preclinical studies is the molecules discovered here, the drug discovered here is checked in vitro, like in cell cultures, and it's also checked in vivo in mainly mice models to see if those drugs are effective or and also if it causes any side effects in these cells or mice. So once the process is successful in preclinical studies, they move on to clinical trials where the clinical trials are conducted in different phases from phase 1 to phase 3. So in clinical trials, these drugs are tested in the patients to see if the effects of those drugs are replicated just like in the animals or in the cell culture. Then once the clinical trial is checked, the drugs go into the approval phase, primarily to organization like Federal Drug Administration (FDA), who approves. the organization which approves the drugs to come into the market. So coming to the next slide, we start to understand in detail. So here we are talking about understanding the disease biology. So when you carefully look into the picture, so it's divided into two. One is the normal cell pathway and the other one is the disease cell pathway. So primarily you have a gene, so the DNA which is the blueprint of life. So this is what gets transcribed into RNA and then gets translated into a functional protein. So when the gene is normal without any disease causing mutation, the protein synthesis is smooth. And due to the smooth protein synthesis, these proteins don't interrupt any pathway. When it goes into a pathway, it gives the cells the normal cellular function like efficient metabolism. So as a result of which the person's physiology is normal, the environment, the organelles are also healthy as a result of this. But once you come into the rights, take a scenario which is where the gene is mutated with the disease-causing variation. So what happens here? So here, the protein which is synthesized from these abnormal genes come tend to are abnormal again. These, if you see here, these proteins are not normal. This is an abnormal protein. So as a result of which it can disrupt a particular pathway. So what pathways can these abnormal proteins interrupt? So it can cause protein aggregation and then it can cause mitochondrial dysfunction. It may even lead to synaptic signaling defect. So this is the problem with the mutated DNA, and this is exactly why understanding the disease biology is important. So in terms of protein aggregation, you can take an example of Parkinson's disease where alpha-synuclein get aggregated. Then there are a lot of mitochondrial disorders which are caused due to mitochondrial dysfunction, and you also have a lot of neurodevelopmental disorders due to these synaptic signaling defects, especially epileptic encephalopathy can be a prime example. So as a result of all this, the physiology of a patient of a person is not normal and he or she becomes a patient here. So the next process of the pipeline is identifying a particular target. So when you look at the left side of these pictures, these are the common targets. It can be an enzyme, it can be any receptors, for example, G protein coupled receptors, which are important for various cellular pathways. It can be ion channels, which are the primary conductor of signals, especially in the nervous system. Then an RNA molecule can also be a target here, or in certain cases, protein-protein interaction can also be a therapeutic target. For example, if two abnormal proteins are interacting, which in turn causes a disease phenotype, then that particular interaction needs to be stopped. So that becomes a target here. So what? So coming to the center of this diagram, of this image. So what is happening here? This is the target protein in cellular signaling pathway. So here you will be able to see different targets here. So targeting this particular cellular kinase here affects a particular pathway, and then once so this is the receptor is another target where a ligand comes and when a ligand comes and sits here, it indeed influences a particular pathway where where it goes inside the pathway and it affects the target protein. So then after this, so what are the technologies for discovery of these targets? So one thing is genomics, where you identify the disease causing variants and biomarkers. Then comes the CRISPR screening. So CRISPR is a molecular scissor which we will come into in the upcoming slides. So this is systematic gene knockouts. A CRISPR screening needs a systematic gene knockouts. Then comes bioinformatics, where you do a large scale analysis of big data. And then comes AI and machine learning, which scientists use nowadays in order to predict the drug target interaction. So coming to the next slide, it is target validation. So in the previous slide, we have identified the targets and now we are validating it. So coming here to this visual, you will be able to see two visuals here. So at the left side, when the target is activated. So what happens when a particular target is activated by, say, an agonist, like a ligand or a molecule? It starts a plethora or a cascade of reactions. So once a target is activated here, the pathway remains functional and there is an expected biological response and a disease amelioration, which means the disease is reduced. Now coming to the right side, when the target is switched off, what happens here? So this leads to another cascade of reaction, but in this scenario, the pathway is disrupted and there is no biological response and the disease state always persists. In other words, to make keep things simple, if you potentially activate a target, there is full signal, which in turn leads to a therapeutic potential. But when a target is off, there is an inhibition of the target. There is no signaling or a disrupted signaling, which in turn leads to minimal therapeutic effects. So what are the validation methods here? Gene knockout, CRISPR editing. So the first two, so in terms of CRISPR editing, this is a molecular scissor, which is a precise genome editing for target control. So what happens here? Scientists design gRNA sequence which matches the DNA in the organism, and the gRNA guides the Cas9 enzyme, which in turn cuts certain nucleotides in the DNA. And what happens here is that leads to a loss of function mechanism. So once there is a cut introduced in the DNA, the repair process start, which is error prone. That is homologous recombination process. So since it is error prone, it leads to a frame shift, which in turn stops producing any uh, abnormal protein. And then you have a gene knockout, which is deleting a specific gene to observe effect. And then you can, they also test it in animal models in vivo in order to see how the knockout works in those models. And if that particular knockout is lethal, then scientists usually stay away from using a particular knockout method. And then biomarker analysis is also done, where the molecules are measured, molecular indicators are measured in order to see what functional changes happen here. So what happens next is a hit discovery. So now we have identified a target, we have validated a target. So now the next process is hit discovery. So what is a hit? So that's a molecule which shows activity, which shows a proper activity against the target. So, what are the discovery approaches of these hits? So, one is high throughput screening, then virtual screening, then fragment based discovery, and then you have it, scientists identify hits through natural products. So, high throughput discovery is an automated process where you have a comprehensive compound library. So after this is processed, there is an automated testing and detection of hits here, and after that the hit molecules are identified. So here, when I say it is automated, scientists always use robotics and also computational based methods in high throughput screening. Then comes virtual screening, where in silico molecular docking is done, which in turn leads to the computational prediction. And there is so once there is computational prediction of various molecules, these molecules are filtered, which becomes the virtual hits here. So the next process, which is fragment based discovery, is so these fragments which you see in this image are potentially weaker molecules. So the what happens? The concept here is these initial small, these weaker molecules are combined together in order to identify binders. In order to identify binders, and then what happens is these are connected and optimized into fragments, which builds leads from the all these weaker fragments. Then comes the natural products, the product discovery. As the name suggests, you can see various natural products here, plants or fungi. So here compounds are extracted, there is bioprocessing done, and then there is isolation of chemicals. So once the chemicals are isolated, those are screened for unique scaffolds. Then after that, these screening extracts are used as hits. So all these processes together are identifying lead compounds for optimization. So this is a multifaceted approach. So coming to the next process, now there is a complete shift here. So where artificial intelligence is used in drug discovery. So coming to the left side of this particular image, you have genomic data, you have structural data. So this is of the protein, then chemical library and clinic data, which are the data inputs which are used in order to find any drugs here. So what happens here, this is the AI pipeline stage, where the first stage is data processing and integration, where the data are clean, filtered, and there is multi-omics fusion which is happening here, that is genomics, transcriptomics, and all that. So once these data are filtered, it goes into training and development using deep learning and algorithms like CNN and RNN. So then comes extraction and learning. There, so in the extraction process, the key biological biomarkers or the biological patterns are studied. Then comes the application specific processing, which is customized model, which then leads to a final output, which is prediction and generation of molecules, of potential molecules. So the applications are basically AlphaFold and generative AI and deep learning. So all these you can see variant prioritization, then protein structure prediction. Then there are virtual screening which rapidly screens a drug candidate. Then there are drug repurposing here which finds new uses of existing drugs. Then there is molecular generation where there are novel chemical structures which are designed. So these are the AI applications here. So next comes the structure-based designs where the core concepts are protein 3D structures and binding pockets are identified computationally, and then in silico molecular docking is done using various tools where they predict interaction of any ligand or molecule with that particular protein. And so the once the docking happens, it gets there is this affinity optimization, that is the hit optimization, which is done in turn to increase the binding affinity of any molecule or a ligand. So, so what are the technologies that are used? So first thing is X-ray crystallography, which uses protein crystals. So once X-ray is passed, there is this diffraction data, which then is studied as electron density maps, which and the final output here is protein structure. And in cryo-EM, the scientists use vitrified frozen data, which is captured using 2D drag particle, which is captured using camera. So so what these camera captures are 2D drag particles, which in turn leads to the 3D map of protein. And then you have AlphaFold. AlphaFold is slightly different because here it is basically working on deep learning networks or deep learning models, which in turn predict the protein structure based on amino acid sequence. Then comes the lead optimization. So what is the important concept here? It's structure activity relationship. It's an important concept here, and with this, what hap- due to this relationship, structure activity relationships, what are the things which improves here? It improves the potency because which means there is increase in biological activity with effective concentration of that. Then comes the selectivity, which make sure that there is no off-target binding of a particular drug, and the drug binds specifically to a particular target. Then comes solubility because the drug has to be soluble inside the aqueous system. And comes the stability, which is pharmacokinetics. So pharmacokinetics is basically the study of how drugs interact with the interact inside the body, and the drug has to be stable because if those drugs are unstable, then they can be degraded by enzymes, which then can deteriorate the disease amelioration. So if you have to ameliorate a particular disease, the drug has to be stable. And the final important process is safety, where the drug shouldn't be toxic, it shouldn't be binding off targets, and it shouldn't be causing any side effects. So next thing is the therapeutic strategies. What are the therapeutic strategies in rare diseases? You have small molecules, which are basically oral dosage. You can see those are kind of tablets, which diffuse through cell membrane and target proteins, specific proteins. Like for example, you have aspirin. Then we have monoclonal antibodies, which do target the membrane receptor or surface receptor, and it binds to the antigens and cause an immunological reaction. The main example is the trastuzumab, and then we have gene therapy, where the blueprint or the genetic material is delivered into the body in order to fix the faulty genes. And the prime example is the Luxturna here. And you also have RNA therapeutics in order to synthesize a new protein, siRNA, which is used in order to reduce a particular disease. And you also have enzyme replacement therapy here, um, which replaces defective enzyme in order to ameliorate a particular rare disease or a defective enzyme. So here we are going to talk about the gene therapy and RNA therapeutics. So gene therapy basically replaces or repair defective genes, and in RNA therapies, antisense new oligonucleotides are used, or sometimes siRNA is used, sometimes mRNA therapeutic is used. So here, as we have spoken earlier in gene therapy, it's a blueprint or the DNA is delivered in order to repair and repair and give a stable gene, which in turn expresses in a stable manner in order to give a new functional gene. And in RNA therapeutic, we have ASOs or antisense oligonucleotides, which binds the pre-mRNA, which blocks the expression or gives alternate splicing. Then we have siRNA, which is gene silencing, which in turn leads to the mRNA degradation via the RIS complex. Then you have mRNA therapeutics, which directly uh, leads to the protein production, which directs the protein production. So the primary example where it is used is spinal muscular atrophy, where ASOs are used, and also gene replacements are used here in order to treat the disease. And then you have precision medicine, where you have to give the right treatment to the right patient in the right time. Um, so what are the technologies which enables personalized care? So one is genomics, then next is biomarker, where you measure specific biological indicators for disease diagnosis and prognosis. And you have AI systems in order to analyze complex data and to predict what kind of drugs can act on specific diseases before going into laboratory work. And the next is multi-omics integration, where you integrate genomics, transcriptomics, and metabolomics here in order to give a proper input of therapy. So next comes the preclinical studies. So what are the key areas? So first thing is toxicology. So where it assesses off-target bindings, and if there is off-target binding happening, does it lead to any side effects or harmful effects or any lethal effects in those animals, dose response and all. The next comes efficacy, where the therapeutic benefit is measured, and in terms of disease amelioration and target engagement. So, for example, if a particular disease is very effective, then it can ameliorate the disease to a very significant extent, which means the efficacy of that particular drug is very high. And then pharmacokinetics, as mentioned earlier, that's the study uh, where scientists see how the kinetics of the drugs work inside the body. So there is understanding, there is drug disposition here, and they see the time course and the bioavailability of the drugs inside the body and how it interacts with various targets to ameliorate the disease. And then you have animal models, you have even in vitro models. So animal models are basically the mice models which scientists use in order to identify the effect of a drug, how it cures the particular disease there before going into the human trial. So that is what this picture of the right summarizes. So here you are assessing the safety before a human trial. Then next comes the clinical trials in rare diseases and the challenges, as we have mentioned before. There are small patient populations, genetic heterogeneity, and limited natural history data here. So, in genetic heterogeneity means there are varied genetic causes for similar symptoms. For example, an epileptic encephalopathy phenotype can have various subtypes, and each of those subtypes are caused by mutations in various genes. So that is why it is heterogenic in nature, and there is limited natural history data where the disease progression is unsure, and due to which the biomarker is hard to measure, and in turn the treatment effect is not properly known yet. So, what are the innovations driving success in rare disease? So, that is adaptive trials, which are flexibly designed, modeling and updated. It's modeled and updated based on the incoming data. Then you have biomarker driven studies, which measure specific biological indicators in order to predict precise outcomes of drugs. And you have cross-border partnerships, which is global cooperation. So what this global cooperation does is, this is one way where you can increase the data sets, you can pool knowledge of patients here. Then next is the challenges, as we have spoken earlier. These are limited data sets. You have high development costs, regulatory hurdles, which means stringent approval process, lengthier process and all. Then you have translation gaps where where there is challenge in moving from lab research into clinical side. So basically, there is a challenge moving from lab to bedside, and there are ethical dilemmas or data privacy which do hinder the research and development of these drug discovery in rare diseases. Then in future directions, there are AI designed therapeutics which people are working on, and multi-omics integration. Then there is CRISPR therapeutics. Digital twins is one of the main aspects which is being talked about everywhere. So the concept here is they create a digital twin on the patient, a digital you say a digital model of the patient where you can where you can test all these drugs, validate these leads, hits, and everything inside the digital twin of this patient. So that's why it is a real-time virtual model which predicts health simulation. And personalized medicine is basically individualized. It looks into individualized care pathways and improved therapeutic outcomes. So basically, the lab is clubbed with the home and patient wearables, like wearables in the sense like smartwatches and all. So it's kind of an integrated ecosystem and patient-centric care, where the prime rider of this process is AI. So, what are the key takeaways? So, here we have molecular dysfunction in rare diseases, where you have rare diseases arise from various molecular dysfunction. Then there are diverse patients and unique profiles. Then you have foundations in understanding biology, and then you have target identification and validation. Then after that you have AI-driven genomics and therapeutic designs, and then precision medicine, as we had spoken earlier. So these are the key takeaways. So then I come to the end of the presentation. Thank you very much for the opportunity. I am ready to take any questions.
So, okay, so we have come to the end of this webinar today and we have come to the end of today's session. Thank you very much, everyone, for your time, and I thank all the presenters here who have presented live and who have sent those recordings for today's webinar. Thank you very much. So we'll be meeting again on 16th May, where there'll be two themes discussed. One of them being foundation of artificial intelligence and the intelligent system, and the other theme will be applied artificial intelligence in healthcare, engineering, and scientific research. So that's it from my end. Thank you very much. Let's meet on 16th May.